Great.
It's going into the MD PhD program.
Oh, great. At Brown.
At Brown.
Yeah, that's fantastic.
How about you guys?
My kids are still pretty young. They're 13 and 15, so they're in high school.
We're good.
Yeah.
We're good.
So
We are good. I'm just gonna test out the wall.
that's great. So is your husband still at Norwood?
Yeah.
Yeah, I was always, I was in awe of how you both seem to do so much professionally, but kind of be there for your kids.
Yeah, it's, it worked out.
Uh-huh.
Mark works very hard to structure his life so that he could be there for sporting events and all events for the kids, really. I think I do get a lot of grief for not being at all the baseball games, so. I can live with it though. It's okay.
Yeah, I know it's
There you go.
it's not the ones that you go to, it's the ones
Yeah.
that you go to.
You look good on YouTube.
Oh, great, thank you.
It's like the dean's events. If you
Yeah.
show up, nobody notices. If you're not there, everybody does.
I'm out.
Great.
The kids forget that I made it to the plays,
Oh, they're gonna hear everything that I
parent-teacher
do.
conferences, the pediatrician appointments. I
Yeah.
don't get much credit for those, but the baseball games, they're big. That's all right, though. It's a revisionist history now, that Mom was never there.
Yeah, they've done very well. So
Yeah, no, they're happy. And,
Yeah, that's
it's been
what's
nice
most
having them close. Jared, the older one, is going to be moving to New York City,
Mhm.
so,
So another reason to come.
yeah, exactly.
Yeah, that's great.
Hi,
Hi.
I'm Jessica with Westchester.
Oh, great.
Thanks for
Nice to
having
meet you.
me. Nice
Oh,
to meet you as
yeah,
well.
thanks for coming.
Yeah, of course. It's really turned out to be a beautiful day. Great event.
I know, it is, I think we got lucky.
Yeah, after all that rain, like for
Yeah.
what, a month straight, I think.
Yeah.
Well, there's thunderstorms in the forecast for this afternoon, so it's not over yet.
Oops, I should have.
Did you want to take pictures of the speakers in front of the second?
Yeah.
Would that
That would
be
be
okay?
great.
Would you guys mind? Okay, and then we'll take a picture of Raj too, maybe even with the speakers.
Yeah.
So I'll go see if I can round up.
That sounds good. I'm just doing some test shots. Feeling out the room a bit.
That's kind of, our local Time magazine is Westchester.
Oh,
So if you can get into Westchester
Wow.
magazine, that's a big deal.
Great. Yeah, it's a big deal. But they're very great. I mean, at what they do, and
Yeah.
they keep it really interesting. Which is
Yeah.
good. And Westchester has lots of authors.
It's true. Oh. Let me see up here. This one up, because I was like, this one looks pretty dirty. Switch. All right. All of these. These are beautiful.
Yeah, really.
Let's see. They look significant.
And they look like the logo,
Yeah,
which is cool.
yeah, it's amazing what they can do.
Okay.
So,
So
so how are
when
your
they're coming in.
neurosportive
Yeah,
efforts?
that's nice.
I think we mostly hear about the BCI stuff, but what about
Shots or single
other
shots
types of
or-
stuff? Because I'm
Okay.
not convinced that's kind of ready for prime time.
yeah, after Lo left us, I have to say, a lot of the efforts that you're doing sort of lost momentum.
Yeah.
This is Jessica, our photographer.
Hi, Jessica.
How are you?
Good, how are you?
Yeah.
These are
So they
distinguished
still
speakers
have
and awardees.
some funding to do,
All right, so
so
let's
let's
do
do
some
work
individual
and
shots.
research that we're doing for
Tell us who we want.
But they're trying to.
Yeah, that's perfect right there. It's perfect.
Yeah.
Running around. No, it's delicious.
Yeah.
Perfect, just like that.
That seems
Lovely.
reasonable.
And then the briefcase keeps going on.
Okay, this is going in the New York Times, so.
It's a little Hollywood here.
Well, we hope so.
That's
Beautiful. Thank you.
Good, good.
Thank you so much for coming. Did you see where you're sitting? Okay.
That's all that iron.
30 minutes. So of course, I wanted to make sure you have your seat. You can, I try to come down, you can comment.
Beautiful. Great.
Okay.
After you.
Yeah, it works for me.
I just go there. I get up at 9:30.
Did you do the one for the anniversary? Yeah.
Thank you.
Well, we did so.
Go ahead. Drop
Yes,
lines.
yes,
Yeah,
yes.
do it. We need some excitement. Remember these one by.
At the 40, yeah, and Guy.
There's a
Nice. You didn't bring your tux? I was gonna wear a ball gown. I was gonna wear a ball gown last night, but I thought it was a little.
Yeah.
Oh, no.
I can never let you
This
dress.
is cute.
I don't think I
I know, he dresses well.
So, Jack, you might like that.
So how are you?
Your prayer.
My dad.
It's gotten a little bit better.
My dad's like, Jack, her brother.
That was great. You know, well, I don't know where to like think of
Okay,
it.
so my
You're all
brother,
looking terrific, but
Get my brother's down for all the.
Okay.
All the day.
Doesn't mean, they may be
Yeah.
home.
I when I tell you like my
They're
cousins
all
are
departments.
surprising, and they're either, attorneys.
So I've done all the calculations, and it's like at least $8 or $10 million a year, and that's conservative. We have, I think now 55 clinicians,
So
about
Uncle Jack is
25.
retired and has been for a while.
Yeah.
Great, thank you. All right, can I give you?
And then was working for
I'm done. It's so hard to keep.
And they're originally from like the mountains,
Why did you leave?
They went to Connecticut, so. They were
Okay.
yeah. So I'm like, yeah, there's like, oh, yeah, every time I go to
You can't
some board event, I run into someone.
So I managed to tell also.
And then all these people I'm like, oh, my dad.
I just lost one of my MS stats in Austin.
And he knew my dad was 33 I am, and I'm working a while, like, go in there with like, sugar and they're like, I was confused, and I was like, how did I know my dad?
Bruce.
So, yeah.
Bruce.
Here you go, Bruce.
He's coming.
He's coming back.
So,
Yeah.
I'll do the ladies first, then.
So
We're gonna bring it in together.
This isn't the full palette of our speakers.
Okay, that's all right. We're just gonna get some group shots while we can. If everybody could shift that way, that'd be great.
Power women.
Oh, I love it. Thank you. Good shot, everybody. Beautiful, thank you. All right, just hang tight. We'll wait for others to arrive.
Yeah, but it's so hard to write anything, it's so competitive to neurology talent, it's hard to find it.
So, Chris, let me introduce you to everybody. So,
Did you know what happened to the
He's gonna show you. First of all, I have a boy.
Yes,
Right.
we see you there here later.
Okay. Likewise. Oh, this is.
Sorry, I just wanted to introduce you. Chris, member of our
Hi,
board.
Chris, I'm Louise.
Hi,
Nice to
Louise.
meet you.
Nice to meet you. Hi. Hi, nice to meet everyone. Well, good luck with the presentation.
Thank you. It's a beautiful venue.
At the beginning, this is an extremely busy time for me. I normally would not show up in jeans.
Oh, I didn't even notice. Those are like dark pants, dress pants. Yeah, they look, you look good, yeah.
So I apologize. I will be in and out, so I'm not, if I stand up while you're speaking, it's not because I disagree with what you're saying.
Excuse me.
Isn't that BS?
I'm still going. No, yeah, thank you for letting us know.
So please forgive me for
What's
that.
the accounting behind that?
Research, exactly.
Return on investment.
ROI, maybe, ROI.
Oh, no, it's the indirect solution.
Oh, the IDCs, I just found out, they get like 87%. I was like,
We get 87.
no, here.
Yeah, but it's, the percentage just reflects the fact that we're spreading it out over a smaller number of investigators.
Right.
So
Well,
the
no,
percentage-
from your NIH,
Yeah,
which
but
the
it doesn't-
NIH,
it's
sorry.
87%.
I guess because of the research institute, rather than having like a hospital with generating revenue or whatever.
We have
I would love that.
15, yeah. So we're spreading all of our indirects over
Across all of them.
15. You probably have 250 at UT. You have probably 500. And so it's not the percentage, it's the
It's
total.
Total IDC
Of-
pool.
Pool.
Yeah.
And I
Ours
don't know
is
why I
like-
can't convince HS. They say, look, your rate is at, I said, our rate is high, but, you know, we have $10 million in
You've got to
NIH
pay
funding.
for that.
Yeah.
So
So you gotta pay it out.
no, but our indirect cost recapture is a drop in the
low.
bucket
Yeah.
compared to.
Yeah, because ours is 57.
So they probably, so
I
you,
think Hopkins is 60. Yeah.
I mean, but that supports all those vice presidents.
And a growing number of licensees.
How many associate and assistant vice? Yeah, we have.
The problem that you have in an academic institution is any administrative duties that are done for a single department are not considered benefiting all the projects in the institution.
Right.
So they can't be slotted into IDC. Where all the sort of committee-related activities of our faculty can be slotted into.
That's
OPM
nice, yeah.
Because
Yeah, we can't do that, benefiting the whole institution.
We
So what
have
you
a
should do-
single ORA pool
Yeah.
that is shared by all the departments as needed, because our ORA, it's just a disaster, and everybody who is good gets recruited to oncology or
Yeah.
ophthalmology that has more money.
Yeah.
So we have a constant turnover of grant support people, and
Yeah.
it's just awful, and we always have openings.
So I think we have the advantage.
Good morning.
But we have the advantage in that we, you know, we don't have to deal with it now, ours.
Which is, yeah.
So I think if you can set up a separate institute,
Right.
that is financially separate,
Then
then
you can negotiate. That's a good idea, actually.
then you have.
Don't
the
do that.
ability-
But UT would never allow us to do that because they take so much of my IDCs. I only get 28% of the 57% back to the department.
Yeah,
And
but-
I have to pay for my own grants people and everything.
Yeah.
It's,
Yeah, but they have to tell you what they're spending.
oh, yeah, they'll make stuff up.
Yeah, they'll make stuff.
They'll
Like
make
the
stuff.
advertising.
Oh, yeah.
And all the
Okay.
power.
Yeah, it's like,
the
okay.
Beeline power.
Yeah, the
It's
room
not
over
funny.
and. I just gave up. I was like, billboards for other people. Yeah.
Wait, so I can't use it anymore, the Beeline power?
Yeah.
So we just need to get.
Actually.
There's a computer bag.
Yeah. Christine, is Edwin out there?
Yes, Ronnie Hopkins.
Yeah.
We're all
Some
done.
boards just, they don't bill
Yeah, we have.
subcontracts.
And I think the photographer, Jeff
Yes,
has.
okay.
Is gonna be taking photos of your work.
Okay.
And here's proof.
That's exciting.
My mother.
How are you?
It's good to see you.
Do you mind real quick? And then we got you one more.
Well, there's somebody here in November.
Right.
January, February, March.
And all the beautiful azaleas, everything's in both weekends is gorgeous in spring.
So we such a read.
That is beautiful here, though.
Well, you know, I have to admit, I was in Florida for the winter, so I have to abstain from that comment.
Yeah, it was.
Just check out.
Hey, Bob, congratulations.
Yeah.
Sorry, let me introduce you to everybody.
No,
I'm Louise.
I can't even tell.
Yeah, we really look great.
I have this folder. And that's where I
I see you wearing this.
Your turn. Your first. Do I have it up there already? I'm not sure where you want to give it to. Oh, you did it on the back? Mine was the only one. Right. Mine was the only one that didn't have a picture on it because that's my last board. So after 14 years, I'm off that board.
So, anybody else?
Everything going on with the rebuild. The company now up for over $300 million in current dollars. On a project that's probably going to take 5 years to sell and 10 years to build. So what's $300 million worth?
Oh, was it really? It was.
I had an honorable sitting with.
All right.
What do you want to do that? Hi, Jessica, Bob.
No, I won't.
Good.
Morning.
Yes, please.
You're right.
Yeah, we're going to.
I mean, I like his
All right.
But of course.
So the catch was great despite looking at her. I was kind of Chris. What's that? So I totally, I talked to John about it. I am incensed that we have people in the building that are disabled and we have no fire alarms working, and no one's doing. So he says, let him take care of that, because I was about to set it off on fire. That's just true.
They also had.
No, that's exactly what I've done. I'm very happy. I have.
These people often have CRD. It reflects the fact that we haven't done so you know, as a management person, that's why I sent the email. Yeah.
And I mean, I'm in better position now because last time was my last hospital board meeting ever. So I can be a little less even-handed when I write. I showed Chris those. They handed these out. As the new ones to run all the buildings. Did you guys get these? Okay, because I don't think the other ones work on the doors anymore.
No, they don't.
Okay,
We got it.
okay.
But I think we're.
Mine was the only one that didn't have a picture on it. I'm sure it expired last night when I woke up the door. I've never been there.
Yeah.
Well, I've kind of, we have kind of worked everywhere, pretty much, but now I don't even know what that is. So do you have a towel on?
Yeah, and so I increased everybody's salary.
It's really nice, right?
Well, I like that. Yeah, good job. And I think it made
Yes, I saw that.
But there's certain things not true because
Yeah, we're in no way.
the rest of the city's quite cheap and there's no paving.
You know, some roads here all day.
Better.
That's right.
So I was
And I do think you take good care of people and see as long as nobody's pushing you. And I actually do all it takes for is my and that was my decision.
So. Well.
That's fair. And what I only do.
So that's why I apologize.
Right here. Right? Yeah, I do. The Saturday market. I could put Abby's on. We could have my. Or you want to put? We can put Abby's jacket. Okay, that works. Now you got the yeah, come here. It's gonna be great. Maybe nice and slow. Perfect flow. And I'm like, okay, all right. I was wondering. Yeah, but you know, they do other events and he's going to do so we'll definitely.
Hold on, that was awesome. At least 3 months want to check the agency.
I know where you are, I have my email.
But I know they, I got on the phone. She said,
Yeah.
I don't know.
Yeah, I'm just in the group. Somebody there. Thank you for your help.
Well, it's,
Well, that's what they do when people really.
Yeah, I put this here because the photographer's going to sit here. We're going to put Bruce and Alex over there. Where are you sitting?
I don't know who will be around. No.
Yeah.
I have no choice.
So she put your job.
Yeah, sit down.
Systematically.
Yeah.
Never, right, then you take.
That's all I got. It is. I was like,
I mean, going up and running, you know, lock yourself up in the bathroom. Pretty much. You know, I feel sorry for her. Every other minute. And man, once I set up the presentation,
What are you gonna do about it?
But I think you all right. I don't. I'm sure so much for coming.
I know, I should have back there.
Hey, how are you? Just responded to an email from Scott. Sent it to footnote to the financials. I know, how are you? Closest foundation.
This is good. Of course, in this campus, there's no better place to be in the spring on a sunny day than this campus. The museum, you're right out, everyone's out. I like to do on all the buildings.
I have to do my job.
Kind of pearl always.
I'm gonna be down
My goal.
You're welcome.
we don't have one of those friends really.
It probably isn't the way we laid it down because of all the wires and places and things or whatever. So one of the questions that came up is the question how many states? So the problem is, we factually representing the possibility because that there is not one person that's possible.
So we don't need to. And I think that when somebody gives us
Even then, they're not sure. I mean, that was the discussion.
And I think you have to.
But my point is, very little worse.
I read it. You don't have to. So it doesn't have to be.
Yes, people think it's honestly, but in reality, playing two things. What I said to the the amount of money not in every they would like sometimes just put it on a piece of paper and then they'll just we have something that makes it reach out just to move the process forward to grow job because otherwise
Three seats right here. How are you doing? No, you're perfectly fine. You're perfectly fine. Nice and comfortable. Okay, so me is not here yet. Yeah, it's like a dark.
5 years to get it approved.
It was here early.
Yeah.
It just is that we should get something on paper.
She was one of the first.
Yeah, but I think that's what we should really do, but I think the aspiration. That's good.
Yeah, she works here, so she's probably, yeah. Okay. Yeah. Yeah, and then I guess, when, if you have a literature chat now, we'll do it after. Okay. Good. Yeah, we'll have time. Yeah. Yeah.
The guys was like here. Like sunscreen.
But I think it's
So it's. He's a trouble, but it's not.
Right, I heard there was a second.
They're likely to be very future.
Yeah, thank you.
No, I'm sitting right there. Just be careful of this because I don't want to hit it and then have to re-center it. That's good. Because I know that I will.
I'm starting to that. So those are the two things. But the foot is able to.
There are 5 people watching.
Well, that's why when they said that, they said somehow he was in third construction.
It's streaming live.
I'm thinking it's a gift.
But we have matching, should have matching. Yes. And the
I like this so much.
yeah, I'm gonna have to look at my slides and figure out which ones we can skip.
Yeah.
Future.
I think we have 25 minutes.
You and I will be very old.
I like it. You could just say, this thing.
But if we were in this
Well, the tripod, yeah. But not the
You're still tired. You're still tired. Because I have to take a break.
Yeah, way the coffee.
That's funny. Well, I was speaking in France, in Toulouse, and they were opening this new neuroscience building.
What are you checking to make sure there's no?
No, I accidentally. And but my talk was during the.
I don't remember hearing, I don't remember a partner.
Senegal.
Oh, forget it.
So like, everybody was, walking out of the store, and I was surprised they was.
And it turns out in Christine and those years, the teacher turned around and introduced the semi-finals. 4 countries that were represented by people in Wow. This is
Really?
Oh,
yeah. What is good?
I take no responsibility for this action.
She's paying me for that. But I'm over here.
Take care of a lot more.
So I only make $40. There's no numbers.
I get hungry out.
You get number two?
Thanks for bringing the key up. Mom has it. I was supposed to go over there yesterday, pick it up.
She only has it I have it in the car with her.
No, it's probably in the house. She was gonna leave it on the counter back in our stuff to clean out. So I was supposed to go pick them up and I forgot about it. I went to that
Only thing I can think of is when you were showing it, and we were turning them. Maybe you turned one off, and you were showing it where it turned it off. Yeah.
But anyway, she said, don't mind me. She said, maybe I confused her with one of the other ones. But she went back the next day. I have the I then realized I don't care as long as it's
I mean, it's under anyways. It's under all the water mains shut off.
No, water ones that you shut off. So with that hot water, I'm waiting for that type of throughout the unit because every with hot water is working with those.
You need to flush it open?
No, because you don't want to get freeze.
No. You know that your cold water shut off, but you still have hot
Maybe able to feel that through their outlet. I think I should just register if you don't mind.
No, they're laughing outlets in this place.
This architecture with things.
I don't think
and I didn't want to you weren't here.
Running itself.
I'd like to give them a kind of introduce them for I work for a college player.
Oh, yes.
Great.
You're there? You're also fine.
It's not coming.
So I have your seat. I have your seat. Is there any slides in here that you
I just
Did you really? Okay. Now next time, snap a picture and send it to us.
All right, I can get everybody's attention.
Third time I've sat.
Good morning, everybody. It's a great pleasure for me to welcome all of you to the second annual McDowell Symposium. I think we were initially worried about the weather, and so we figured that, the old saying is one umbrella should keep the brain away. We figured 180 in a row certainly keep the rain away. And we're fortunate that that actually turns out to be the case, because we have this beautiful day. I know it's a little bit of a tease to actually have to sit in here, but I think you're going to find it a real treat. We have a really exciting symposium planned for you. We're also here to enthusiastically honor 3 McDowell awardees, and we're looking forward to doing that as well. So the theme of our symposium today is hope for stroke. And I think for those of you, there are many people in the audience today who really conceptualize what that means, but just to give you some daunting statistics around this clinical problem, there's 1 stroke every 40 seconds in the United States. That means that before I finish my introduction, and I promise I'll be brief, there will be 22 new strokes in the United States. There are 800,000 people every year who suffer a stroke in the US, and there are over 7 million people who are chronically disabled from stroke. One of the aspirations of this institution is to be able to really harness the best in contemporary neuroscience at all levels to really repair the brain and spinal cord. But ultimately, our success can't be judged just by new discoveries or simply by NIH grants or papers in great journals. Our success is going to be defined by whether we're successful at the bedside, actually in humans. And I think that is, so I think it's really important to understand that part of the reason why we felt it was really appropriate to have this event as part of the McDowell Symposium was because that was an abiding goal of Fletcher's from the time he started the Institute, and even before that. He did some of the first trials of PT and stroke. He did catalyze some of the first use of robotics for stroke. He started the journal Stroke, which is now the leading academic journal in this area. He started the National Stroke Association. He had an enormous investment, and if that wasn't enough, the problem took on a tragically personal turn in that his wife was suddenly passed away from a hemorrhagic stroke at the age of 55. And I think that this really prompted him to redouble his efforts and really focus on how cures can be achieved along the spectrum, whether that's prevention, acute treatment, or repair. So I think that we've been really inspired by Fletcher's legacy, and his daughters, Lynn and Laura, are here today to celebrate his legacy with us and really have helped catalyze our focus on stroke today, which I think you're going to be really excited about. I will finish my introductory remarks by telling you that we realize, importantly, that we're not going to be able to solve this problem alone. And part of our motivation in bringing together this incredible group of speakers is really to give you a sense of some of the exciting successes that are occurring in the field, but really to have this be a collaborative exercise with everybody in the room and the speakers to really identify critical gaps that we need to be focusing on as we move forward. And I will say that you're going to have an opportunity to ask questions of our speakers, both after they speak, but also during lunch. And I think that it would be a missed opportunity if you don't go up to each and every one of them and find out what they think how they think we can be doing better, because clearly there is lots of room for improvement. I also think it hasn't gone unnoticed that all of our speakers are women, and they have been remarkably successful, not only as leaders in the stroke field, but also in leading departments. And they've surmounted considerable cultural and institutional challenges to reach remarkable heights. So the other thing, I think particularly, we are a training organization, and I think particularly for the students and postdocs and junior clinicians who are looking to get advice about both men and women about how to really overcome some of these real barriers. I think, again, I would encourage you to touch base with them. So before I introduce our first speaker, I wanted to thank Christine and Rebecca and Liam, Pat, and the rest of the committee that put this incredible day together. And you can see the beautiful tent outside where we'll have lunch. I wanted to thank them all because there's an enormous amount of time and effort that goes into planning an event like this. But I also wanted to thank all of you for taking time out of your very busy schedules to actually make it to the Burke campus and join us. There'll be opportunities for tours in the afternoon, and I'm hoping at lunchtime that I'll be able to share with you some of the details behind a exciting new initiative that has been funded by the state to set up a clinical trials network in New York for chronic stroke. So hopefully I'll get a chance to tell you a little bit more about that at lunch. So without further ado, let me introduce the first speaker, because I think you're anxious to hear from all of them. Dr. Lauren Sang-Sing, who's the head of the stroke program at Yale. And Lauren is a remarkable physician scientist. I think she's doing some amazing work which allows her to understand what's happening with the immune system, both inside and outside of the brain, not only in models of brain hemorrhage, but also in human disease. And she just joined our scientific advisory board, and I think we've had an opportunity over the last several months to get an opportunity to talk with her about how we can synergize our efforts with the great program that she's actually developing at Yale. So, Lauren, you're up.
Good morning, everyone.
Good morning. Got the microphone on?
Yeah, I don't think I can get your copy.
Can everybody hear me? Yes. All right, good morning. Thank you, Raj, for the very kind introduction and the opportunity to address all of you today. It's a pleasure and a real honor to be here at Burke. I'm constantly impressed with the science that's here, and it's wonderful to have got to know many of you and hope to get to know many of you more. So today I'm going to be speaking about bridging the gap, patient aids and research innovations for intracerebral hemorrhage. I think it's not surprising that the worldwide incidence of ICH has continued to increase. In this global burden of disease study that was published a few years ago, looking at data from 1990 to 2010, we saw a 47% worldwide increase in the first ever incidence of intracerebral hemorrhage. Now, there were in 2010, about 5 million hemorrhagic strokes, and that includes both intracerebral hemorrhage and subarachnoid hemorrhage. And about 80% of those are in low and middle income countries. And so the burden of this disease is disproportionately shared in our countries with the least resources. I'm going to be focusing today not on all hemorrhagic stroke, but really on intracerebral hemorrhage, which is caused by a rupture of the blood vessel within the brain, generally a small penetrating arteriole, that leads to a mass of blood in the brain itself. And so here's just a schematic, and here's a CAT scan of a patient that we had recently at Yale. And you can see this massive blood in the brain that's obviously not going to cause a lot of neuronal injury. About 15% of all strokes, when people say stroke, about 15% of them are ICH, or intracerebral hemorrhage, and that's in Western countries. It is responsible for about 40% of all stroke-related deaths. And there are about 2 million ICHs, particularly worldwide. ICH has an incredible morbidity and mortality. While about 60% of all ischemic stroke patients, about 60% of all ischemic stroke patients do recover some functional independence long-term. That rate is only about 10 to 20% of ICH patients. And mortality overall has remained steady over the past decades at 30 to 50%. So I think of ICH as a process. There's the initial event, the rupture of the penetrating arteriole, often caused by hypertension, but also by vascular abnormalities or cerebral amyloidopathy or other causes. And then there's a period of hematoma expansion and hemorrhage growth. We know that about a third of patients are still bleeding when they present to our emergency room. And this hematoma enlargement is causing further tissue destruction, white matter injury, and shear forces. We then enter a phase of inflammatory responses. And this is where I become very interested, because we know that there's robust cytokine and chemokine production. So those are the chemical signals that cells make to call on the inflammatory response. We have activation of the innate immune cells of the brain, called microglia. And we have robust recruitment into the brain of cells from the peripheral immune system, particularly in the early days, neutrophils and monocytes that then become macrophages. We then have a process of the lysis of red blood cells, toxic injury from iron, activation of other neuronal cell death pathways, and then a resolution phase in which the hemorrhage is cleared out of the body by phagocytes, the cells that gobble up debris, and the secretion of growth factors that hopefully will aid in neurological recovery. What we haven't known, particularly in humans, is what is the time course, and what are the mechanisms in which these pro-inflammatory, pro-injurious pathways switch to these more reparative pathways, and what are the signals that actually drive that response. So I think about it as this time course, this acute injury phase, this subacute immune phase where there's robust leukocyte recruitment, and this resolution phase. And we're really focused on those transitions between inflammation and resolution. We can do this by modeling in mouse, by two different models. A blood injection model in which we can control the ICH composition. So we stereotactically inject blood into the right striatum. We're limited a bit in size because if you inject too much blood or too rapidly, it just refluxes up the needle injection tract. It's a little bit technically complicated. We can also inject collagenase, which is an enzyme that degrades the basement membrane and leads to bleeding in the brain. This is a little bit easier for us to perform. It causes more injury, certainly more robust inflammatory response, and a longer recovery phase. But it does have some limitations also, particularly that the post-translational modifications are bacterial. So when I was starting my lab, now 9 years ago, I set out with one goal, and that was to simultaneously study these processes in humans and in mouse models. And in order to, the reason to do this was to have continuous insight from our patients to drive our mouse models and what questions we could ask from our mice. Now, mice are flexible, right? They're easy to use, they're easy to quantify, we can vary different immune responses, we can take advantage of genetic flexibility, but they don't always model what's happening in our patients. And so we want to continuously validate our pathways that we find in mice with human data. And this requires us to have access to patients and engage patients to consent and provide their biological samples so that we can learn directly from them in the recovery, the inflammatory process, and the recovery phase. And so we do this by launching sub-studies of large clinical trials of hemorrhage of vacuous. We did this through the MISTY trial, we did it through some other trials, and then also through prospective biorepository and prospective patient cohorts to get cerebrospinal fluid if there's an external ventricular drain placed, or peripheral blood samples that we can look at their monocyte and neutrophils. I'm going to focus on two main populations and really try to give you a little bit of a flavor. I'm not going to get into the weeds too much of data for this talk, but I will show you a couple of different stories that have come out of my lab. To set the stage, we have two different macrophage populations in the brain after injury. So in homeostasis, we have those resident immune cells, the microglia, and they continually self-renew. They're here in blue within the brain. And then we have bone marrow-derived monocytes that circulate in the blood. And after brain hemorrhage, those monocytes are recruited to the brain and become macrophages. So in the rest of the talk, if I say macrophage, I'm talking about those cells that were recruited from the blood, enter the brain, differentiated, and now are inflammatory cells in the brain. And if I say microglia, I'm talking about the resident cells. And we can differentiate these cells by flow cytometry. So this is just a standard flow cytometry plot where each cell, each dot here is a cell that we've harvested from the brain. And depending on which cell marker you're using, you can isolate T cell populations, neutrophils, macrophages from microglia. And from a typical mouse brain in the perihematoma region, we get about 20,000 microglia out and about 3,000 blood-derived macrophages. And then we can phenotype them and ask, what are these cells doing in the brain? What happens when we deplete them? Are they contributing to injury or recovery? And so one of these populations that we tried to deplete and ask those particular questions were macrophages. So we took an understanding that macrophages are very pleiotropic. They can be very pro-inflammatory or they can be very reparative, and asked, well, what's the time course for them being injurious or reparative? So we used a macrophage depleting antibody for the first couple of days after we created a hemorrhage in a mouse, and asked, well, what happens to the mouse behavior? Because phenotyping a cell or understanding what a cell is making it has value, but understanding how a mouse is functioning when that cell is present or absent has more value. What we found is that across a whole number of different tests, if we took away those macrophages, the mice were much better off during the first couple of days after hemorrhage, indicating to us that those macrophages in this pro-inflammatory state were injurious to functional outcome for the first 3 days. If we let the macrophages come in for the first 3 days in this time scale, and then started depleting them during the recovery phase, we found the opposite. That the mice that did not have macrophages during the subacute phase had more hemorrhage left and failed to recover on these tests compared to the mice that had normal macrophage recruitment. So this highlighted a dual role for macrophages in the brain. First, they're injurious, and then they're reparative. So that's how to take a step back. We then sorted macrophages out of the brains of mice at all different time points and asked, well, what's happening? What genes are they upregulating during this recovery phase? This starting to come on at day 3 and then highly upregulated at day 7. Compared to what's happening in that early phase, what's coming on at day 1. What we found was that day 1, not surprisingly, were classic pro-inflammatory mediators. But in the recovery phase, the macrophages were upregulating pathways that were really heavily involved in the clearance of the hemorrhage, the efferocytosis, the gobbling of cells that have died within the brain. And here's just a volcano plot of some of those differential gene expressions. So efferocytosis is the way that our body's natural immune system clears cells that have undergone a type of cell death called apoptosis. And there are a number of receptors that are involved in this process. We focused on a couple of them, AXL and RTK. And what we found is that if we activated macrophages with thrombin here, we can get them to be pro-inflammatory. But if we fed them cells that were apoptotic, those cells that within the brain need to be cleared, they downregulated their pro-inflammatory pathways. So we have a model in which macrophage is inflammatory. We see these red blood cells from the hemorrhage within the brain that are expressing a particular signal, phosphatidylserine. The macrophages eat them, gobble them up to clear the debris, and now they become more pro-reparative. As I mentioned, we're very tied to human data, and so everything that we find in a mouse that we find interesting, we then try to validate in human cells. And so we created human monocyte-derived macrophage cultures and stimulated them in the same way, fed them those apoptotic red blood cells, and asked whether we can induce the same kind of change in the cell phenotype. And indeed, we found very similar findings. Those cells had to be eaten by the macrophages, and those red blood cells had to be giving that signal, that phosphatidylserine signal, in order to induce that change. We then wanted to know, well, in our patient cohorts, does this matter? Does this correlate with outcome? And so here's just some data from culture that showed that with stimulation, the cells are actually cleaving off these receptors. And we can detect these receptors in the serum of our mice. And the higher the circulating level of this soluble factor AXL, the worse the disability was in the mouse. So then we looked for this soluble factor in our patients in a prospective cohort. And in this cohort, it has classic IC volumes and show scale scores and other severity measures. We also found that in addition to those severity markers, higher levels of this soluble AXL in the blood predicted for recovery out to 1 year. So something that was happening at day 3 in our patients, this cleavage product, was predicting recovery out to a year. So we have this model in which macrophages are coming in from the peripheral blood, becoming activated in the brain, contributing to injury, and then in the process of clearing the hemorrhage are becoming reparative and are important for functional recovery. So here's our 2 macrophage populations. I showed you data on this, where this process of efferocytosis and the clearance of the hemorrhage leads to resolution of inflammation. I didn't show you data, but it's been published, in which the microglia use a very different signaling pathway, not mediated by TGF-beta, for them to become pro-inflammatory. And this produces, leads the microglia to produce neurotrophic factors, growth factors like BDNF. So far, and I hope I've convinced you of, is that inflammation contributes to early injury and then is critical to functional recoveries. We don't want to block inflammation. It's really important to allow pro-recovery inflammatory processes to go on. So we have to be much more nuanced in understanding the pathways that switch a pro-inflammatory or pro-injurious pathway to a reparative one. And we can and should study these diseases and these processes in both mouse and in humans, because eventually it's the humans, our patients, that we want to cure. Now we're lucky in intracellular hemorrhage because there is a surgical option, and there's a lot of interest in minimally invasive surgery to remove the hemorrhage, to see if removing the hemorrhage early would improve functional outcomes in our patients. This was recently tested in the MISTI-3 trial, a worldwide NIH-funded trial looking at the efficacy of minimally invasive surgery using a catheter placement approach to improve outcomes out to 365 days. We launched ICHC, the clinical trial, that collected the brain evacuated each day and a matched peripheral blood sample from 16 sites, patients enrolled in 16 sites across the United States. We have data on 22 patients that were in the surgical arm. So we have peripheral blood and brain hemorrhage evacuated, sorted for different cell populations that we were interested in, and a temporal course from the initial placement of the catheter and then samples that were collected on each day for the next 5 days. Here's what the workflow looked like, and I'm going to show you data on the neutrophil population, both in the brain and the peripheral blood, and the monocyte macrophage population in the brain and the blood. So from here on out is unpublished new data that hopefully will be published soon. We started out by just looking at what is the spectrum of the immune response in the brain compared to the blood. So this is principal components analysis. So we just looked at the entire transcriptional profile of both the macrophages shown on your left and the neutrophils shown on your right. And what principal components analysis does is looks at the entire variability and the entire spectrum of activation. What genes are those cells turning on? And then looks at how similar or how different are those activation pathways across all the samples. And it collapses that difference into this sort of 2-dimensional plot. Principal component 1 captures the most of the variability. And so factors that drive principal component 1 are accounting for most of the variability. What you see here in yellow are the hematoma monocyte-derived macrophages compared to peripheral blood macrophages, monocytes in our patient's blood, and then a cohort of age and hypertension matched controls that we gathered peripheral monocytes from. The fact that the peripheral blood monocytes and the healthy control monocytes are very close together indicate to us that not a lot of monocyte activations happen in surgery compared to the vast amount of activation that's happening in the hematoma-derived macrophages. And we see a similar thing in the neutrophils, where principal component 1 really separates what's happening in the peripheral blood from what's happening in the hematoma. So most of the activity, most of the action is happening in these cells once they've entered the brain. We then were able to define the time course of activation. So looking at hematoma-derived macrophages and hematoma-derived neutrophils. And we found that there were basically two different clusters of gene sets that were being differentially activated over time. There's a gene set that was being highly upregulated over the first 90 hours, and a gene set that's being highly downregulated. And interestingly, that change is all happening in those first couple of days, and then the gene profiles level out, at least during this time frame. Interestingly, the genes that are going up and down in neutrophils follow the same kind of time course, but actually most of the genes are quite different between the two cell populations. Interestingly, the number one driver of what is happening in those cells when they're activated in the brain is glycolysis, the way that the cells are using glucose to produce energy. And that's true in both the macrophages and the neutrophils. It's been known in the immunology field for maybe the last 5 to 10 years that the pro-inflammatory state of the cells is highly dependent on the metabolic pathways that the cells are using. And it's thought that more pro-inflammatory cells are using glycolytic pathways compared to the more homeostatic cells that are using the oxidative phosphorylation and citric acid cycle. So we have this model that a macrophage utilizes glycolysis, so it becomes very pro-inflammatory. Or it sees these anti-inflammatory signals and downregulates glycolysis and becomes more reparative. So we asked, well, is that happening in our patients? So if we just look at that early response, we see robust, so very red, high upregulation of many chemokines, cytokines, and encapsulants. Simultaneously, we see very strong upregulation of glycolytic pathways and downregulation of the TCA cycle and the pentosphosphate cycle. We then asked what's happening over time, because through MIST we have the benefit of this temporal response. And we found that over time, the cells are downregulating glycolytic genes in both macrophages and neutrophils, while at the same time, they're downregulating inflammatory markers and upregulating hemoxidase 1, a very important antioxidant and anti-inflammatory mediator. So with this data, now we're analyzing, well, what are the transcriptional profiles that are associated with good outcome in our patients? And how can we then take this information back and model it in our mouse to test candidate therapeutics? And I'll say in parallel, we're now moving to a single cell level and looking at single cells within the hemorrhage and within the peripheral blood that allow us to identify new populations and novel activation states that have never been discovered before. So I hope I've convinced you that it is possible, at least in ICH, to move seamlessly between hemorrhage, patient data from the hemorrhage and peripheral blood and mouse models. And we're moving towards utilizing observations that we're making from patient samples back into mouse models, back into patients, understanding the pathways, looking at therapeutic target testing in mouse, and then understanding the temporal windows from our human data. So in summary, this resolution of inflammatory responses is critical to recovery. It's regulated by tissue-specific mechanisms. So what resolves inflammation in the brain after hemorrhage is different than what resolves inflammation in the brain after ischemic stroke or after other organs. We can use complementary animal models in human studies to understand how these processes are regulated. We think that macrophage metabolism is emerging as a very important therapeutic target. And understanding the biology in human patients is critical, especially when these pathways are changing rapidly. And with that, I'll thank the people in lab, Wawa, Jaydong Chen, who's now a faculty member in Taiwan, drove the atherosclerosis and macrophage modulation work. Mike Askanase, who's pictured on the right side, who's driven all the ICH-seq work and transcriptional profiling work. We work closely with Chris Long and Alex Shalik up at MIT in Broad, and Britt Goods, who's the postdoc up there that we work with. Of course, Dan Hanley, the MISTI trial, and the BIOS team for helping facilitate the work. Our ongoing collaborations with Ron and scientists here at Burke, the MISTI patients and other patients who have consented for giving us samples to allow us to understand the disease in the hopes of developing new targets, and of course, all the funding that makes it possible. And with that, I'll take questions.
So, Ed, do we have a microphone for questions? Can we use the microphone?
Are there questions that anybody has? Maybe I could start with a couple. So in terms of therapeutics, is it rational, given these sort of distinct roles of the macrophages acutely and then the reparative phase, is there a way to think about, for instance, modulating soluble Axel pharmacologically that allows you to define what's the right time actually for the therapeutic?
I think Axel is complicated because it's regulated both transcriptionally, and so we see this robust activation of production of cell surface Axel, but then in the inflammatory state, it's cleaved. And so perhaps inhibition of cleavage would be a rational therapeutic target, and we're thinking about ways to do that. It's cleaved by adamantine and 17, and so those mechanisms are known and could potentially be modulated. I think what we're understanding is that maybe it's not also a very macrophage centric, of course, but I think many of the other mechanisms that what our neutrophil is doing may not be regulated by the same pathway, but the metabolism may actually be the common link between switching of immune cells from pro-inflammatory to pathology. So we're very focused now on how do we modulate that metabolism, and how do we change a cell, how it utilizes energy to become more prolific.
I wonder if you could tell us about your tissue in MISTY. So I presume it comes from surgeons trying to get into the center of the clot and remove it.
It is.
And sometimes they actually were successful, but sometimes they were mystic. But anyway, but I'm wondering if these changes are going on within the central part of the clot, or really going on the edges where we are trying to spare axons, or in the case of a basal ganglia thalamic hemorrhage, trying to spare gray matter components.
It's a great point. So indeed, we know, because we have the CAT scans in all the patients, we know exactly where the catheters were in MISTY. And because we were getting samples out and blood was draining, we know that these were engaged within the hemorrhage. But we are particularly measuring what's happening in the center of the hemorrhage, which might be very different from what's happening in the periphery. There are white matter tracts that are going through the center of the hemorrhage that ideally we'd like to spare as well. And I think what we know from comparing the mouse data to the human data, that a lot of what we're finding in the center of the hemorrhage is indeed also happening, at least in the macrophage and neutrophils within the sort of perihematomal region. What we don't get out of MISTY, but we will get out of some of our other studies, is what's happening in CNS resident cells. Because we don't see microglia or MISTY evacuates, and we don't see astrocytes, and mostly not neurons, a couple of small populations. And by utilizing some other tissue sources, including brain biopsy specimens and autopsy specimens, we're now able to do single cell nuclei-seq, so single cell nuke-seq on fixed tissue specimens will allow us to get to that perihematomal region and understand what's happening in the other CNS resident cells. But we need to be creative about those approaches and how to get those tissues.
Great.
Hi, Laura. So first of all, I think we're all amazed at the amount of work you've done. This is really fantastic for the ICH community. My question is about MISTY, and as you know, the trial itself didn't hit its primary endpoint with respect to removal of the hematoma. So the question is, what's the role for actual hematoma clearance in mitigating these pathways? And is it your hypothesis by the time you were able to get into the OR and get these catheters in, that the inflammatory response was already deregulated and has such impact on outcome that actually removing the clot at that point is more or less, the point? Or because intuitively you would think that if you get rid of the exciting stimulus for the inflammation, which is the clot, that the patients would have done dramatically better. So I'm just wondering if you could comment on sort of that bit.
It's a complex question. I think the exploratory analyses in MISTY, which were pre-specified, but indeed not the primary outcome, show that if the surgeon was successful in removing the clot below 15 ml, that there was a benefit in terms of patient outcome. So I think it opens up a lot of questions about whether or not we are whether our technology isn't there yet in terms of safe and efficient removal of hemorrhage, whether the time period was right, was it too late, was it too early, was the horse out of the barn? I think what we see is that there's very robust early activation, which would suggest to us that maybe if we could safely and effectively get the clot out earlier, that might benefit patients more. But I think that's quite speculative. And I think using some large animal models and using some other trial design through that field to answer that question. I think what Missy did show us is that it was safe. And so there's a path forward to then start to ask these questions in very well-designed, rigorous trials.
Maybe one other point I think you've made to us is that the MISTI data that you've analyzed is surprisingly tight, isn't that right? That you would have expected it to be really noisy, and it was surprisingly clean.
So I think what as the PI embarking on a study, and especially a transcriptional profiling study of human patients, we had no idea what the patient variability was going to be, what our success rate was to be able to get high quality samples from 16 sites around the country. We were unsure about whether we would be adequately powered to answer some of these questions. And what I think you're saying, and the data show, is that the initial stimulus to brain hemorrhage seems to be so powerful that the patient-to-patient variability in these samples is minimal compared to the overall activation state of what happens in the brain. And we see these very tight curves of what's happening for many pathways, not all, but many pathways that really give us insight into the biology. And then we can ask sort of later studies about different patient factors that might drive responses.
Great. Thank you so much. Any other questions? Lauren's going to have to leave early, so I think you'll have to email her with additional questions.
But please do. I'd be happy to discuss more.
Yeah. Go ahead, Gary.
How do you visualize using metabolism as a target when the brain itself is so dependent on metabolism and all the other tissues are? How could you use it to shift these cells selection?
It's a great question, and one that we're asking in the mouse model now about whether we can give systemic agents to modulate cellular metabolism that would penetrate the brain, reach our targets, reach our cells of interest. We're not trying to inhibit metabolic pathways, but the robust activation of glycolysis seems to be quite specific to activated pro-inflammatory cells. But I think we are very limited in our understanding of how to target some of the metabolic pathways at this stage, but are very interested, and I'd be thrilled if you had ideas and we could talk more about it.
Great. Thanks so much. So our next speaker is Dr. Karen Curie, who's chairman of the Department of Neurology at the Warren Alpert School of Medicine at Brown. And she's also a neurologist in chief at the Rhode Island Hospital. And I've known Karen had the pleasure of knowing Karen for a long time. She was head of the stroke service at the Massachusetts General Hospital. And as many of you may not know, I think the general agreement in the neurological community is that stroke was discovered, if not invented, at Mass General by one of Karen's predecessors, C. Miller Fisher, who said we learn neurology stroke by stroke. Karen has really taken on the torch from C. Miller Fisher and has done really elegant work on the prevention of stroke, on acute stroke, and has been very much involved in understanding how we train clinical neurologists to actually do research. And she's been very important in a variety of different NINDS and NIH committees that really are allowing strategic directions that offer exciting new opportunities for the field. It's great to have you here.
while he's bringing that up, I want to thank Raj very much. It's a pleasure to be here. I've heard about your institute for many, many years. And it's delightful to be here on this gorgeous day. Just driving up and seeing all the young people and the energy is very inspiring. So I'm looking forward to the rest of today's program. So my talk is more in the clinical realm. Thank you. Lauren is a very tough act to follow, and she certainly provided a vision for how the work done in the preclinical setting is going to help us better understand intracerebral hemorrhage, both in pathophysiology and for treatment possibilities. But I'm going to switch gears a little bit and talk about stroke prevention. I'm sure that everybody in this room would agree that given the choice of having good therapies to treat stroke or being able to avoid it altogether, we would choose to avoid it altogether. And in fact, there are several good strategies that can be implemented to help prevent people from having stroke. And so I'm going to talk through those for a bit in the next half hour and give you a sense for what you can do and what you can share with others, your friends, your families, your patients, to try and reduce the burden of stroke in the U.S. These are my disclosures, none of which are particularly relevant. So Raj brought up some of these statistics in his opening comments, and they're in the booklet that you have in front of you. And the reason that we all include these statistics in our talks is to remind everyone of how common stroke is. This is not an unusual diagnosis. This is not a rare disease. And it's a very scary one because 80% of people who have a stroke do not have a warning. They're literally normal 1 minute and perhaps paralyzed next minute or unable to speak or to see. And that's a very frightening thing. In 20% of people, you have some prodrome, a TIA is what that's called. It gives you some indication that you're at risk, but 80% don't have that. There's also a sense it might not happen to me. Why? Because I'm young. Stroke is a disease of the elderly, and that's not true. And unfortunately, we've seen sort of the reverse, which is that as younger people develop poor eating habits, sedentary lifestyle, and vascular risk factors in their 20s and 30s, they're having strokes in their 40s and 50s. So they're moving stroke up by several decades compared to the usual pattern. In addition, many people have had silent strokes. So a stroke, when we talk about it conventionally, means someone experienced a symptom. But in fact, you can have blockages of blood vessels that go on asymptomatically. And it's estimated that 13 million people in the US have these strokes, so that if you did an MRI scan, for instance, you would see evidence of it, but they're not aware that they've had an event and they don't know that they're at risk for symptomatic stroke. And therefore, this is a tremendous public health issue. Only 20% of stroke survivors can't live independently. They can't go back to work, they can't go back to driving, they can't go back to living a full routine life. And that's why the Burke Neurological Institute is so important. We need to do more to help people with stroke so that they can get back to full independence. Lauren already brought this up, but I wanted to just reiterate it, that when we use the word stroke, we're really talking about three big entities. The first is ischemic stroke. Okay? That comprises the majority of strokes, and that's really what I'm going to focus on in my presentation. In this problem, a blood vessel gets blocked, and as a result, the tissue downstream doesn't get energy, doesn't get blood, doesn't get oxygen, and it dies. So you can see that in the cartoon. My pointer's not working, but the gray area is the brain tissue that has died as a result of not getting blood. And on to the right, the top CAT scan shows that same thing. That whole area that's dark on the right side of that image, that's all dead brain. And that's dead because the blood vessel was occluded and
I don't think it'll work.
Oh.
Not on this side.
No, oh, he's right.
Yeah, maybe you could try the computer.
Just a mouse, maybe?
Yeah. Okay. So this is the dead brain on the cartoon, and this is the dead brain on the CAT scan. Now, in contrast, The kind of stroke that Lauren was talking about is intracerebral hemorrhage. That accounts for somewhere between 10 and 15%. That's blood in the brain itself. And then the last type is subarachnoid hemorrhage. And that's the kind that's commonly caused by an aneurysm that ruptures, and that's blood around the brain in the subarachnoid space. So how do you prevent stroke? And how can we change things, whether they're risk factors or lifestyle factors, in order to reduce that risk? And this is a pretty exhaustive list. It's probably not everything, but it is sort of the big things. That if you have high blood pressure, heart disease, atrial fibrillation, high cholesterol, diabetes, blockages of the carotid arteries, a previous stroke or TIA, some abnormality in your heart structure, migraine or sleep apnea, you're at risk. And probably if I polled my expert colleagues in the first couple of rows here, we could come up with a few more. But these are really the major ones. The behaviors are not negligible, and I will talk about some of these. But cigarette smoking, sedentary lifestyle, obesity, and alcohol abuse are also very common contributors to stroke risk. And while I'm not going to pretend it's easy to change any of these things, particularly in individuals who are stroke prone, it's critical to prevent the inevitable stroke. I'm going to start with hypertension. There are two reasons I always start with hypertension. The first is the prevalence. It's incredibly common. 25 to 40% of people in the US have high blood pressure. So this is again, speaking from a public health or epidemiological perspective, a very important risk factor. And the relative risk. How much does it increase your risk of having a stroke? 3 to 5 times. And so even though all of these are important risk factors, hypertension is the biggie. And this is probably the most frustrating risk factor because we know it's critically important to preventing stroke, and yet it is so hard to get people to get it measured and have their blood pressure treated. So there's a lot of confusion about what an optimal blood pressure is, and that might contribute to people not being as aggressive as they should be. So the most recent categories defined by AHA have normal at less than 120/80. Less than 120/80. 120/80 used to be the normal, but now the goal is to get it even less than that if it can be tolerated. Elevated blood pressure starts with anything above that. And I sort of wish that we could all now take out our blood pressure cuffs and measure it because I think it's dramatic. And I bet many people would leave here surprised that their blood pressure was not meeting these goals. And then as your blood pressure goes up from there, you go to higher stages of risk and higher stages of hypertensive urgency. But what has really struck me over the course of my career is that what we considered normal and what we thought we had to treat has drifted down. When I started, it was at 140/90. And then it drifted down, and there was even a statement that had it up as high as 150 for a brief period. And now we're all down thinking less than 120/80. And neurologists have sort of always known this as opposed to cardiologists, because every study that looks at this question shows that lower blood pressures are better for the brain. I want to show you a cartoon about the small blood vessels in the brain that might become more obvious why that's the case. So if you specifically ask, with regard to blood pressure, how much does your risk of stroke go up? If you're over 120, your risk of stroke goes up 26%. And if your blood pressure is in that 130 to 139 range, your blood pressure, your risk is almost doubled, or more than doubled. And you can see the incremental increases as you go further up. So in an ideal world, we would all be sitting here with systolic blood pressure less than 120. But that is very, very hard to achieve, even in people who don't have risk factors. So high blood pressure is very insidious because it doesn't cause that stroke over the course of a day or a week or a month. But when the small vessels in your brain see that higher pressure on a relentless basis over months and years, the vessels start to be damaged. And so this is a cartoon of the brain. It shows the large vessels that bring blood up from the heart to the brain. And then, just like a tree branches,
the blood vessels in the brain branch, and you get the small twigs that go deep into the brain. The small twigs are very easily damaged by high blood pressure. And over time, they get thickening, and they get clots that form in them, and the small vessels block off. Sometimes it's silently. You can see it on an MRI, but the patient doesn't know that they're experiencing it. But if you're unfortunate, one of those small vessels might be feeding a motor pathway. And when it blocks off, you suddenly become paralyzed, face, arm, and leg. And there are several parts of the brain that are particularly susceptible to this: the deep structures in the hemispheres of the brain stem, and the cerebellum. And so as a vascular neurologist, we frequently see people who come in with a symptomatic stroke in this area, but also more extensive damage that we can see on the scan. And just to demonstrate that to you, I brought an actual example. This is a patient I took care of last week. This is an 80-year-old man with poorly controlled hypertension. All of this white stuff that you can see in the brain, all of that is damaged, scarred brain tissue as a result of those small blood vessels being blocked over time. This patient did not have any focal symptoms related to that, but had memory difficulties and walking difficulties. And just as a nuance, the brain is also shrunken. And I wanted you to see by comparison an MRI of a normal brain that doesn't have all that white matter scarring and doesn't have the shrinkage that comes when the brain gets scarred like that. And so we frequently see this. This is not an uncommon finding in people who have high blood pressure that hasn't been well controlled. We also know that very small improvements in blood pressure translate into big benefits for stroke reduction. So if you can decrease the diastolic, that's the lower number of blood pressure, by only 6 millimeters of mercury, you can reduce stroke by 40%. That's huge. If you can reduce the higher number, the systolic blood pressure, you can reduce risk by a similar magnitude, 36%. And even in the elderly, for those who think, well, if you live to be 80, what does it matter? In fact, reducing blood pressure, even in the elderly, has a significant benefit in reducing stroke risk, and therefore improving healthy brain aging and quality of life late in life. This is data from the PROGRESS trial, which was a clinical trial that compared blood pressure reduction to placebo. The active arm of this study used a combination of an ACE inhibitor and a diuretic. And as you can see, there was a significant reduction, 28% reduction in stroke risk in patients who were taking the ACE inhibitor and the diuretic. So this study was a positive study. It demonstrated that blood pressure reduction helped prevent stroke. But it's important to walk away from that study and realize that although this trial used an ACE inhibitor and a diuretic, any drug that is successful at bringing down blood pressure will achieve similar benefits. And so reducing the systolic blood pressure by only 3 millimeters of mercury, a very marginal difference, can reduce stroke risk by 10%, and again, that reduction in diastolic blood pressure by 40%. So no matter what type, what strategy, what mechanism of antihypertensive, if you get the effect, the patient benefits. Atrial fibrillation is also a major cause of stroke, although the mechanism is different. In atrial fibrillation, there's an irregular heart rhythm that's caused by a disturbance of the pacemaker in the left atrium. As a result of the abnormal beating of the heart, there can be sludge that develops in the left atrium, particularly in the appendage. And so clots begin to build up in this area of the brain here. As the heart pumps, those clots can break off, they travel up the aorta and up into the blood vessels of the brain. And they ultimately lodge in one of the vessels, one of those large vessels in the brain, block blood flow, and cause the stroke. So different mechanism. Atrial fibrillation goes up with age. These are multiple curves of atrial fibrillation, the prevalence by age, globally. So this isn't a problem only in the US, it's a problem worldwide. And you can see that the inflection point seems to be people in their 60s. That's really where the prevalence of AFib starts to increase dramatically. As the population in the US ages, more and more people are being diagnosed with atrial fibrillation. Currently, the lifetime risk for developing AFib is 1 in 4 for those who are over age 40. If you look at the prevalence, it's about 4% if you're over 60 and 9% if you're over 80. But as our population ages, this is going to become a bigger issue for public health and for stroke prevention. The risk of developing atrial fibrillation is not only linked to age, it's also associated with risk factors. So for instance, if you have hypertension, you are more likely to develop atrial fibrillation. Diabetes, heart disease, congestive heart failure, and valvular disease also increase the risk of developing atrial fibrillation. So if you take a step back and you say, AFib causes stroke, how can I prevent AFib? We're back to talking about the risk factors that I've been discussing. If you have atrial fibrillation, your risk of having a stroke may be different based on these other risk factors. So what physicians typically use is something called the CHADS-VASc score, and it's an online tool. Anybody can look this up. And what you do is you take somebody who has atrial fibrillation and you ask these simple questions. Do they have heart failure, high blood pressure? Are they over 75? Do they have diabetes, a previous stroke, any type of vascular disease? Are they in that age range of 65 to 74? And are they male or female? And you can come up, you just add it up, because for all of these, you either get a 1 or a 2. And then based on that score, you can calculate the risk of stroke annually. And as you can see, if your CHADS-VASc score is low, if it's 1, your risk is very low. If your CHADS-VASc score is 9, your risk is 15% per year. So you're somebody who's at significant risk, and a multi-pronged approach is probably critical for reducing that risk. Unfortunately, one of the biggest changes in managing AFib in the last 15 years has been the development of new forms of oral anticoagulation. The standard classic treatment for preventing those clots from forming in the left atrium was Coumadin. Coumadin was a very difficult drug to use. It required blood tests to try and keep it in a therapeutic range. There was an interaction between Coumadin and many types of food, so you had to follow a special diet and avoid healthy foods like green leafy vegetables. The newer drugs, dabigatran, rivaroxaban, apixaban, and edoxaban, are all much easier drugs. They're dosed once or twice a day. They do not require blood tests, and they don't have that interaction with other medications and diet that made Coumadin so problematic. The doses don't have to be adjusted. It's one dose for all. So it's made it much easier to use. There are a variety of options. You have probably all seen television commercials for these. There's a lot of direct marketing of these drugs. But for physicians, there's also a logic that can be applied for why one drug might be better for an individual patient than another. The trials all show, and I'll just explain this plot briefly, if the point is on this side of this hashed line, it means that the newer oral anticoagulants are better than Coumadin, and if it's on this side, it means that Coumadin is better. And as you can see, in terms of preventing stroke and reducing risk of bleeding, the newer drugs are, in almost all of cases, better than warfarin. And even those that fall on this side of the line cross null, so they're not statistically significantly better. So having drugs that are as good or better than Coumadin for preventing the clots and have lower risk of bleeding has really been a major game changer. And so doctors can now choose between these various agents and maybe tailor it somewhat to individual patients. So some drugs have a better safety profile. They were associated with less bleeding in the clinical trials, and those are drugs like apixaban and edoxaban. In a patient who has a low risk of bleeding and a high risk of stroke, dabigatran might be the best option. And for people who have other comorbidities, again, there are drugs that seem like they may be safer and more effective than others. It's important for me to point out at this juncture that to date, there haven't been trials that compare these new drugs head to head. So what we're doing is extrapolating from trials that compare the new drugs to Coumadin and then looking at the details of those studies. It'll be interesting to see in the next few years if we actually have some head-to-head comparisons so that I could make these statements with more confidence. When these drugs first came out, there was a lot of hand-wringing. What if we use these drugs and a patient has a hemorrhage into their brain as a result of it? Anything that prevents clots increases risk of bleeding. And when these drugs were first released, we didn't have reversal agents. And so there was a risk. We knew how to reverse Coumadin. We did not have easy tools to reverse these new drugs. But again, that has changed. And so in the last few years, there are now drugs, idarucizumab and andexanet, that are readily available in emergency rooms to treat people who are on these drugs who come in with life-threatening hemorrhage or need surgery. So this has been an important evolution of managing patients with atrial fibrillation that has helped us increase the number of patients actually receiving therapy and helping to reduce the risk of cardioembolic stroke. Diabetes. Diabetes causes stroke through multiple mechanisms. So for one, it damages blood vessels directly and causes atherosclerosis. Atherosclerosis occurs in both large and small vessels of the brain. And obviously, if the pipe is rusty, it's more likely to get clogged and blocked and cause stroke. But in addition, diabetes causes other problems in the body that can lead to cardiac injury and cardiac failure. And so diabetes also increases the risk of developing cardioemboli and atrial fibrillation and causing stroke through that mechanism as well. So treating diabetes is very important for reducing stroke risk. In terms of how much that risk is, these are multiple trials that looked at the risk of stroke, both ischemic and hemorrhagic, in people with diabetes. And for the most part, whether you're male or female, black or white, having diabetes roughly doubles or triples your risk of having a stroke. We know that it is hard sometimes to regulate blood sugars, and that being very aggressive can sometimes be dangerous in that it can lead to low blood sugars, which are not good for the brain either. But from this UK prospective diabetes study, they were able to show that for every 1% drop in hemoglobin A1C, which is sort of a snapshot of how well blood sugars are controlled over time, if you can reduce the hemoglobin A1C by 1%, you can reduce the risk of stroke by 12%. So this is an important piece of information to feed back to patients, that if they're adequately managing blood sugars, they are also reducing their risk of having a stroke. But just like hypertension, there's the issue of, well, should we have to wait until someone has overt diabetes, or is there an opportunity to start earlier? And so the notion of insulin resistance has also crept into our thinking. When you start to show evidence of glycemic dysregulation, might there be a time to act then before diabetes occurs? And I'll talk more about that in just a few slides. So obesity is directly related to this concept of insulin resistance. Obesity can be measured by several ways. I think the most commonly that's performed when you go to see your primary care physician is going to be body mass index, but you can also use waist circumference, waist hip ratio, or waist to stature ratio. And what this all gets at is the notion that a body that is apple-shaped, where you have large abdominal mass, big belly, that's a dangerous type of fat that increases the risk of cardiovascular disease, as opposed to the pear-shaped pattern of obesity, where you have a narrow waist relative to your hip. Obesity has a lot of intermediate pathologies associated with it, like sleep apnea, high blood pressure, atrial fibrillation, all of which contribute to stroke. And fat tissue is metabolically active tissue. It actually increases inflammation. It has an effect on lipid metabolism, and it can directly affect the health of blood vessels throughout the body. And all of that leads to vascular injury and atherosclerosis. So it is not benign to have obesity. In terms of the degree of risk, 30 is the threshold for obesity based on body mass index. And as you can see, if you're up in that range of 30, your risk of ischemic stroke goes up 50 to 80%. However, it's interesting to note that it's not quite as significant for hemorrhagic stroke. A different mechanism, perhaps a different disease entity in this regard. The other interesting thing is that just being obese alone is not the true issue. Despite the metabolically active lipid and adipose tissue, if you adjust for high blood pressure, diabetes, and cholesterol, the risk goes from here, which is increasing, to down here, which is a much flatter line. So it's the obesity causing the risk factors that are causing the stroke. So it gets us back to root cause and how to prevent that. So insulin resistance, this state of prediabetes, directly damages the blood vessels through all of these mechanisms. It also promotes inflammation, thickening of the blood vessel wall, and it activates the cells to promote clotting so that cells in the blood stick to the wall of vessels. And this starts to occur in patients who have obesity but have not yet developed diabetes. I was involved in a trial of patients who had a stroke, and we measured to see whether they had insulin resistance, that's the IR. And as you can see, if you were a body mass index of 18 to 24, a relatively small proportion of people had insulin resistance compared to those who are overweight or obese. So the larger your abdomen, the greater your risk of having insulin resistance. And you can bring down your insulin resistance and improve sensitivity to insulin through weight loss, diet, exercise, and drugs in extreme cases, particularly for secondary prevention. So if you're exercising for 1 hour 3 times a week, you can improve your insulin sensitivity by 13%. So these are small things, but if you add them all up, they can have a significant impact. The message on lipids is pretty simple. HDL is good. That's eating a more vegetarian diet, deriving fats from nuts and olive oil. And LDL is bad, and that's all the junk food. I won't even list them all. Statins are widely used in the US because, unfortunately, first of all, some people don't want to correct their dietary habits, but in addition, for genetic reasons, there are some people who will not achieve good levels of cholesterol despite a good diet, and they need something else. They usually need a medication. In the Sparkle study, being treated with a statin, atorvastatin, reduced the risk of stroke by 16%. And so atorvastatin, or one of the drugs in that class, have become pretty standard therapy for people who have stroke, especially those with the small vessel or atherosclerotic types of stroke. And this shows that atorvastatin in the Sparkle trial reduced not only the risk of stroke, but also major cardiovascular events. So it helps blood vessels throughout the body. Carotid stenosis is a risk factor for stroke, but I wanted to focus on people for primary prevention, asymptomatic patients. The question is, how would you even know you had blockage of your carotid arteries? A lot of times it's detected because a doctor will put a stethoscope up to the neck and hear a rushing sound that indicates the vessel's blocked, or there may be testing done for other reasons, and they do an ultrasound or CTA and find it. This is what carotid atherosclerosis looks like. It's not very pretty. Plaque is sort of nasty. It has calcium, fat, blood, inflammatory cells in it. And on angiogram, you can see it causes the blood vessel to be squished just like a hose. And if you screen just a general population, these are people who came to a mall, a very high percentage of patients will have asymptomatic carotid stenosis on screening, particularly if they have a blockage, if they have risk factors rather. The The blockages can cause particles to break off. They go up to the brain and cause a stroke, or just like a hose, you can just restrict flow and you get low flow strokes. Carotid stenosis is more common in men, and it is also more common as you age. And currently there's a very large trial underway that are comparing 3 different strategies for dealing with carotid disease. One optimizes medical management, tries to fix all those risk factors. One does a surgery called a carotid endarterectomy where they remove that plaque. And the last one is a stenting arm where they put a stent in the vessel to restore flow. And so we'll know in a few years which of those 3 things is most useful. Dietary issues is something I counsel my patients with at almost every visit. These are the recommendations from the American Heart Association. The 2 diets which have the strongest scientific data to support them are the DASH diet and the Mediterranean diet. But they all have very similar features, and that is more of a shift toward fruits and vegetables and getting away from carbohydrates mainly. And the more that you can achieve that in your diet, the better off you'll be from a cardiovascular perspective. It also happens to be a diet that's good for reducing cancer and for other conditions. But it does require a significant commitment to altering lifestyle. And sedentary lifestyle is also something that I frequently counsel patients on. The American Heart Association recommends 150 minutes of moderate aerobic activity or 75 minutes of vigorous aerobic activity every week. And I'll be the first one to say I'm guilty of not consistently meeting these goals. But these are things we should all take to heart and at least be aspirational and maybe try to work toward it and work it into our lifestyles. So if we do all of these things that I've discussed and stay on an aspirin, if your doctor has recommended it, you can reduce your risk of stroke by 80% or more. And that is an incredible feat and critically important for a healthy aging brain. And with that, I will stop. Thank you.
I think we have time for one question. We're running a little late. Bruce.
I've always had a bit of cognitive dissonance about these studies where we're trying to reduce stroke risk by using a drug. And hypertension is a great one. I mean, you look at the studies going back almost 40 years, and you see a decrease in the systolic pressure of 3-5 millimeters, and an effect on risks. But we're aiming so low. I mean, you're still running, average blood pressures are running in the 130s, 140s.
Absolutely.
Why do you think, with the power of the drug companies, trying to get their drugs out there, why aren't there studies trying to get blood pressures into the around 120?
Well, there are trials trying to do that. Sprint just wrapped up, and that was trying to reduce blood pressure for cardiovascular and for cerebrovascular disease. But it's extremely difficult to do. It requires a regimen of multiple antihypertensive drugs in most cases. And it's extraordinarily difficult to get people to do the lifestyle modifications that might actually help sustain them in that level. So people don't want to take 3 drugs. They don't. And they don't want to have to come back for the frequent monitoring. So I think that the solution to this is implementing lifestyle modification earlier so that you don't develop high blood pressures to begin with, and to be aggressive. So once you start antihypertensive therapy, not to be content with the 140/90 and to aggressively get people down into the lower range. But it is not easy to achieve, and very few patients actually hang in there. Even in trials where a lot of effort goes into compliance and trying to keep patients in the studies, is very hard to do. In real life, people just give up. It's expensive, they don't like the side effects, they don't want to do it.
Great, thank you for So I think I just wanted to amplify this point that Karen made so effectively, and that is that stroke is frighteningly progressive and democratic in its afflictions. And I think you'll hear a lot today about different populations of individuals and how they differ. And our next speaker, Louise McCullough, who's the chair of neurology at the McGovern Medical School at the University of Texas in Houston, and the neurologist in chief at the Memorial Hermann Hospital at Texas Medical Center. She's been really at the cutting edge of understanding how stroke presents is different from a pathobiological standpoint and presentation standpoint in women and men. And her work preclinically has had a profound impact upon NIH standards of actually how we do some of our preclinical tests and how they should be done both in male and female animals. In addition, and I think it would take me a long time to go through all the great things that she's done. As chair of neurology at UT Houston in a relatively short period of time, she's really elevated that department beyond its historical outstanding reputation in stroke to really one of the leading NIH-funded departments in the country. So it's great to introduce you.
Thank you, Raj. Okay, and I'm a pacer, and I don't care if anybody interrupts me, and if we don't get through things, it doesn't matter. So if you have a burning question, stop me. I'm going to rely on Raj to give me a heads up when I'm like 20, 25 minutes in. Otherwise, I will talk forever, okay? And actually, I was going to talk about age-related dysbiosis and age-related inflammation and how we can alter that to affect stroke and Alzheimer's. But actually, last night, I started thinking about the audience, and I also wanted to go back to my love, which is stroke in women, trying to figure out why women respond differently to stroke, because it is becoming a silent epidemic of elderly women. Many more women die after their stroke. They're more likely to be put in a nursing home. They're more likely to have cognitive deficits, and there's a lot of reasons for that. So I'm going to just take you through some of it. I also do some basic science stuff, and we'll get into how we can model these diseases in the laboratory. Luckily, Karen, you did a great job, so now I don't have to go through too many of my slides, but there's a quiz on the next slide to see if you guys were paying attention. All right, so types of stroke. We've already seen this a bit, right? So this here is a intracerebral hemorrhage. What is the most common cause of this disease? What's the thing you can prevent? Hypertension, yes, okay, blood pressure. And there's an old Chinese proverb that says, when you're thirsty, it's too late to dig the well. And that's kind of the way I talk to my patients about hypertension. You don't want to wait till you're 70 or 80 to start treating it. The damage is done. So we have to get very aggressive in midlife when we start seeing these increasing prevalences of high blood pressure. But Karen knows, I know, I'm a stroke neurologist. It's hard to talk people into taking 4 drugs, especially because how do you feel when you have hypertension? You feel just fine. Unless your blood pressure is super high, you don't have any symptoms. So I'm going to tell this man that you're going to be on beta blocker, which may cause you to be impotent, and I'll give you an ACE inhibitor, which will give you a cough. So, like, you know, it's a hard sell. So you really have to try and understand why they're not being compliant if they're wrong. Okay. This is a subarachnoid hemorrhage. So instead of the blood being inside the head, inside the brain parenchyma itself, it's outside. It's in the surrounding area around the brain. That's called a subarachnoid hemorrhage. Does anybody know what causes a subarachnoid hemorrhage? Not vascularology.
Blood vessel burst?
A blood vessel burst from an aneurysm. So that's exactly right. So it's an outpouching, like a ballooning of the vessel, and they can be rapidly fatal. Many people die before you get to the hospital. The only reason I bring this up is because this disease is more common in women. It's about 65%. We know it's related to estrogen. We're not quite sure why. This seems to be sex neutral, although as women age, they get more high blood pressure as they age. And then the vast majority of strokes you've heard about are ischemic. So this is a CAT scan. I'm going to give you a quiz later. So CAT scans. This is bone. So what's bright on CAT scan is either blood or bone, calcium. So this, you don't see any white. Well, this is a little bit of calcium here. But this is the abnormality. Do you guys see that? That hypodensity? That is a large middle cerebral artery stroke on the left side. This stroke is going to be devastating. It is going to leave this person with aphasia, the inability to communicate or understand speech, and being paralyzed on the right side. We have got to prevent this type of stroke. We heard a lot about atrial fibrillation from Karen. Atrial fibrillation becomes more common in women, and they also, if they're not on those blood thinners, are more likely to throw a clot like this to a big vessel. Now, the reason I'm spending a little time on this is because, you know, people have abandoned stroke. Oh, we can't cure stroke. We can't treat stroke. And now we have had a game changer. Over the past few years, we realized we can absolutely treat stroke. So I'm talking a little bit about acute stroke here. Not so much prevention, but what happens when you come in and you are paralyzed on your right side? What can we do for you? And now we can do a lot. We can often leave the hospital completely normal within a day. Okay, so I'm going to just tell you a little bit about that. Okay, so the epidemiology of stroke, I got to harp a little bit about women in stroke because it's important to me. So 1 in 5 women will have a stroke in their lifetime versus 1 in 6 men. Interestingly, in most age groups, stroke is actually more prevalent in men. And as you may hear from Dr. Eichler later, when you have neonatal stroke, boys do much worse. They're ischemia sensitive. They're more likely to have neurological problems. They're more likely to have intellectual problems, physical problems than girls. Girls seem a little hardier. We've actually been able to model that in the laboratory. If you take cells from an XX mouse, which is a female mouse, and you put them in a dish, and you compare them to XY cells that are from a male mouse, and you give them what we call stroke in a dish, which is oxygen glucose deprivation, the female cells are hardier, and they die a very different way than the male cells. And this becomes important because if you're trying to design neuroprotective agents, you want to make sure they work in both XX and XY, and sometimes they don't. And that has been one of the problems with translating therapies into patients is we don't pay enough attention to sex. Okay, things change with age. There's very complex interactions, which I won't talk about unless you're interested. You can come and talk to me. Aging, hormone, and chromosomal sex. Like I mentioned, more women are disabled after their stroke, require SNF placement. They have more cognitive decline, more depression. But they also are much more likely to live alone. So if you have minimal deficits, but you can't get up 5 steps, and you have 5 steps to get into your house, guess where you're going? You're going to go to a nursing home because you don't have somebody that can help you. Okay, all right, so we're going to start with a case. And I just highlight this case because it also reminds me to tell you about stroke warning signs. That's important to me. 85-year-old, recent loss of longtime husband, so she's widowed. She was found by her daughter in the afternoon, who visits her every week, but spoke to her the day before. So whenever you find somebody down on the floor, they call me to the ER, they say, oh, we found this woman down in her kitchen. The first thing you want to look for is the F, focal. Is it focal? Because she could have had a pulmonary embolism or a heart attack or pneumonia. But if you see that she's moving one side and not the other, that suggests a focal finding, which tells you an area of brain has been impaired. So that FAST, this is something you all need to know. And I know not all of you are physicians, doctors, scientists. Act fast. That's face, arm, speech, time. Because we can treat stroke. We have clot busters that have to be given within 4.5 hours. And we have catheter-based interventions where we can snake up a catheter through the femoral artery and yank out a clot. So face, arm, speech, time. If you see asymmetric facial drooping, arm weakness, trouble understanding or producing speech, you call 911. You don't call your spouse, you don't call anyone. You call 911 when you get to the hospital. Okay, so her exam showed left-sided weakness of the face, arm, and leg. She has a gaze preference, meaning she's looking away from her hemiparetic arm. She has a thing called neglect, which you guys, it's basically when you no longer kind of understand the space and you can actually hold up somebody's arm and you can say, whose arm is that? And they'll say, it's yours, because they don't even recognize their own limb. It can be difficult to rehab. So where is this stroke? We know when you have neglect, it's a big vessel, okay? So that's a cortical sign. This is critical in our new world of stroke. You need to figure out, is it in a big vessel? Why? Because you can get to a clot in a big vessel. You can get a catheter up there, and you can take that clot out. So here is her scan, and you can see here, this is her CAT scan. It's got the red arrow sign, so that should be helpful for you. But this is some early infarct changes, okay? It doesn't look great. This looks like it's at least 6 or 10 hours old. And you see this little thing here? That's the clot. So she has a large clot in her middle cerebral artery. That's a clot we can get out though. And this is the game changer. So this is a different case. This is actually a left MCA. And here you can see here, this is an angiogram, and this is the clot right here. And then you go in with a catheter right here, and then you take out that clot. So this whole vessel that supplies like a third of the brain is now open. And this patient left the hospital the next day. They came in globally aphasic, meaning unable to speak, and hemiparetic, and they left the next day with no deficits.
What was the time between unblocking?
So this unblocking took about 10 minutes. The sooner you get to the ER, the better. So this basically, we all knew it worked. But in 2015, there were multiple trials showing that the number needed to treat to have a good outcome is 2. It is one of the most powerful therapies in medicine. The number needed to treat for lipids, for example, or using a statin to prevent a stroke is hundreds. 2. So if you treat 2 patients, one of them is going to have a good outcome. The biggest thing is time. That's why fast is so important in getting to the hospital. Because if you already have that brain damage, you can't get that brain back. Once the brain is ischemic, and you guys feel free to stop me. We'll get to the gut later, but if we don't get to it, it's fine, because I think this is important for you guys. So this is the clot that came out of unblocking that artery, okay? Now, what happens to this 85-year-old lady whose CAT scan looks a little concerning? It looks like a lot of that brain is already dead. So now what we can do is do a thing called rapid or perfusion. So basically you get an IV and you put a contrast in, and you see how much of the brain is already dead. Meaning if you open up that blood vessel, all you're going to do is reperfuse dead brain, and they may actually bleed. You may do more harm than good. So there's a thing called a core, and that's the tissue that is not going to come back. It's beyond salvage. And then there's penumbra. You can see she has a massive stroke. She has a lot of tissue at risk, this whole hemisphere. But unfortunately, she has an almost 100 cc core, meaning that tissue is not coming back. She is not eligible for therapy. She was probably down on her kitchen floor for 6 or 8 hours, and this is what happens with a lot of our elderly women. So her clot's here, and you can see that whole hemisphere is dark, and she ended up having a large stroke, and actually she passed away. And what's important with these strokes is understanding, as Karen mentioned, atrial fibrillation. You might not have noticed, but when she said about the Chad's vast, you get a point just for being female. Why? Women are more procoagulable. They're more likely to throw a big emboli like this when they're in AFib. And this is shown here. So the frequency of strokes due to AFib in women, it's like 45%. In men, it's about 20%. They have more atherosclerosis, more carotid disease. And why does that matter? This is a cardioembolic stroke. It's huge. These strokes are big. They shoot up. They block a big vessel. They cause a lot of damage. Versus an MRI showing a tiny little stroke here, this is more likely to be a woman, this is more likely to be a man. And that's part of the reason the epidemiology shows that women are going to do worse. More likely to be disabled, more likely to end up in a skilled nursing facility, more likely to have post-stroke cognitive disorder, more likely to have post-stroke depression. But interestingly, she's much more likely to have functional deficits before her stroke. And now we're understanding that women are at specific high risk because they don't have their strokes until the age of 72 on average, whereas men are about 67. So they have a lot of comorbidities like frailty, and that's hard to control for. So if you look at this, so now we're recognizing the epidemiology over the past couple of years, this is that confidence interval of 1. And if all you do is adjust for stroke size and for age, women look like they have a significant disadvantage. They're much more likely, 35% more likely to be dead at 1 year compared to men, okay? So you can see here, more likely to be dead. But if you adjust for things like pre-stroke disability, like using a cane or a walker, women are actually 20% less likely. The problem is women have more disability before their stroke because they're older, and it's hard to just control for all that. Also, there's a lot of psychosocial factors. Depression and psychosocial stress increase the risk of instant stroke, and more so in women than in men. We don't know why this is. Is it because if you're depressed or you live alone, what if you have like macular degeneration? Maybe you can't get to Walmart or Walgreens to get your blood pressure medicine. Maybe you're depressed, maybe you're drinking, maybe you're smoking. So there's a whole bunch of factors that go into these studies in patients. So we've tried to move it into the lab. We know that social isolation, it has to be subjective loneliness. You have to feel lonely. Lots of people live alone and don't feel lonely and have great social networks. They're not at risk. But if you feel isolated, you have a much higher risk of death from stroke, cancer, many other diseases. We also know it accelerates cognitive decline in those who have been diagnosed with Alzheimer's. Importantly, 52% of women, that's amazing, right? Census data, 52% of women live alone compared to 29% after the age of 80. And most of that is because women are more often widowed. So it's very difficult with compliance or people taking their meds. So we decided many years ago, actually when I was at Hopkins, where I met many of my colleagues that are giving talks today, we decided to model this in the laboratory. So this is the stroke model we use in the lab. This is called the middle cerebral artery occlusion. This is a very kind of common model. It's very reproducible in our hands. It's where you take a suture and you put it up here through this blood vessel in the mouse, and it causes a pretty good sized infarct. This white area where there's no blood flow, that's infarcted. And here is just a section of a mouse brain showing this over time. TGC staining stains the brain pink. So everything that's alive is pink, everything that's dead is white. And I show you this for a reason. This is the infarct evolution over 7 days. So in the beginning, the first hour, you see a lot of subcortical infarct, and this is what happens in humans too. By about 24 hours, and certainly by 3 days, the infarct is mature. This is all stroke in a mouse. And you can see it's not a bad model for the stroke that we see in humans. There's, it's a mouse. But now, really interestingly, if you take females and you separate them, these are C57 black mice. They're all exactly the same, genetically the same, been fed the same, and all you do is separate them for 2 weeks before you do that induced stroke. Look what happens. This is a pair-house female. Here's their stroke. This is one that is isolated for just 2 weeks before stroke. The stroke is so much bigger. So this is biology. This is not compliance. This is not you taking your medicines. This is biology. These are female mice, all the same. Now, this female does horribly. More likely to die, really bad behavioral deficits, but you're like, well, no kidding, Louise. Look at how big this stroke is compared to this stroke. It's not rocket science to say this mouse is going to do a lot worse. Well, they did. So what we decided to do to control for the infarct is wait until 3 days after stroke to separate them. And that way we can control the infarct size. So any behavioral deficits we see are not a result of that bigger stroke. And at 30 to 90 days, this is a socially isolated animal. This is a pair housed with a stroke partner, and this is pair housed with a healthy partner. So a healthy mouse in with a stroke mouse. And all of their infarcts were exactly the same, okay? But look what happened. This is the isolated mouse. 30 days, just we have to stop the experiment. They all died. And this is not the stroke. The stroke is mature at 3 days. So what's going on? Why are these animals continuing to die? They also, if you put them in little interaction chambers, they run to the corner, they don't interact at all, they kind of huddle up, they have all these features of depression. So we're trying to figure out why that is. We think it has a lot to do with microglial activation, and we have a couple target microRNAs, but I didn't want to get into the weeds with it. But there's something really profoundly detrimental about isolation. Okay. So switching gears for the second part and last part of the talk, don't worry, I won't talk until next Wednesday. But I can. So, all right, so.
20 minutes.
Oh, 20 minutes. Oh, we got plenty, 10 more minutes. So neuroimmunology is becoming a very robust field. You heard a lot from Dr. Sansing about this. It's basically the bidirectional communication between the immune system and the nervous system. We used to think the CNS was an immune privileged site, meaning we have a blood-brain barrier and you can't get any cells in there. Well, that's nonsense. Cells go in and out all the time, and in fact are educating us. They're involved in cognition, they're involved in antigen sampling and clearing the brain of debris. So it's not completely immune privileged. Inflammation is, we're now recognizing, plays a role in many different diseases, including aging. So we started looking at the systemic effects of stroke. Stroke has profound effects in the brain, but interestingly, it also has profound effects in the periphery. So basically, this is a thymus, this is a mouse, and this is a sham animal, and this is a stroke animal. So the thymus, which is a repository of T cells, our important immune cell, becomes totally small and shriveled up after a stroke. This is a mouse spleen, same thing, sham stroke. This is something noticed by Keith Pennypacker many years ago, that you give the brain an injury and the spleen contracts to 50% of its size. Why is that? We now know it's neurally mediated and it dumps out all these immune cells because the brain is saying, we're having a challenge here. Now, I spent about, I don't know, like a month looking for the thymus in aged animals. I couldn't find it. So it turns out with aging, our immune system is so different. And that's when I realized we had to move our models into aging. Because I think a lot of the information we're getting from young animals at 12 weeks, which is equivalent to a 10-year-old, that immune system is totally different than the person who's at risk for stroke, which is a 7-year-old. So I started moving into aged animals, and I found the immune system was totally different. There is no thymus. There's very little thymus in humans. All of our T cells have already been educated. So also looking at what Karen had pointed out about a normal versus an abnormal scan. This is a normal MRI of a 32-year-old. This is one of my patients who actually has cerebral amyloid angiopathy, a form of dementia. And you see all this white matter disease, these strokes, huge amount of atrophy. So we have to be very careful. What are we modeling? Are we modeling stroke in this brain, or are we modeling stroke in this brain? So we started doing this many years ago, and at first, when we first started doing this, we couldn't keep the animals alive. They just died. So we started looking at the brains, and this is actually a young male mouse. See how big this stroke is? In an aged male, the stroke is paradoxically smaller, but they have horrible outcomes, higher mortality, and more neurological deficits that they never recover from. Very similar to patients. And if you look at patient data, older patients also have the smaller strokes. They have less edema. And we don't know why that is. So then we started trying to figure out, well, why are all these old animals dying? And we found that stroke led to profound weight loss, and that the aged animals never recovered from that weight loss. And they also had a lot of markers of sepsis. Their blood pressure and their temperatures were low, meaning they were getting infected. So we started saying, okay, well, if they're infected, let's try and find the infection. This is a blood agar plate, and you see these little dots here? These are colony-forming units. They're bacterial colonies. So after stroke, in age, you see how many more of these bacteria. This is a culture from the mesenteric lymph nodes. Those drain the gut. The gut was really distended after stroke. So you get a stroke, and the gut gets distended, and these bacteria cross into the circulation, and especially so in aged animals. Then we noticed that we took these little colonies, we scraped them off the plate, we sent them for sequencing, and we can identify the type of bacteria. And it turns out the bacteria in an old mouse were totally different than the bacteria in a young mouse. So we're like, well, that's kind of weird. They were much more pathogenic. So what we did then is we started looking at the gut, because they were gut-derived bacteria, as a source of these. Now this is a piece of gut, the outside of the gut. These are young animals, these are aged animals. They have been given a gavage of FITC, which is a fluorescence dye. This is the outside of the gut, and you can see in the aged animal, the FITC just leaks right on through, right into the periphery, and it carries those bacteria with them. So we started looking at how the gut looked. This is a normal looking gut. This is where the bacteria reside here, and this is the host. This is mucus. Mucus is a great barrier. It makes it so the bacteria can't get in and give you infections. The bacteria are all out here. These are these crypt cells that produce mucus, so a nice mucus barrier. Look what happens with aging. The crypts start getting really kind of not so great, and look at the mucus barrier. But even worse, look at what happens after stroke. Ooh, that's not good. There's no mucus barrier anymore at all. All of the host is now exposed to all of the bacteria. The host also has what we call a hypoxic barrier. The villi of the epithelium inside our gut are hypoxic, which also prevents bacteria from getting into the host. And this is what happens after stroke. There's no hypoxic barrier. So the bacteria feel, it's like, come on in. So this is a thing called a fish. This is a stain for bacteria. So again, this is the colonic epithelium in a young male animal. This is the mucus barrier, which the host produces and the bacteria can produce. And this keeps us separate. Like, we should not be touching. This is what happens in an old animal. This isn't even a stroke, but you can see the mucus barrier is tiny, and these bacteria are like touching us, touching our host, right? What happens with stroke? Here come the bacteria. Here they are, okay? And in an aged stroke, they're all over. So this is what's happening after stroke. So the microbiome is that bacterial community that lives within us. And we started sequencing the biome, and you can see that the old biome and the young biome are completely different. The old biome has more of these firmicutes, which are more deleterious. So what we said is, well, what happens to the biome after stroke? And what we see with the young biome is it looks pretty healthy. But if you give a stroke, the post-stroke young biome starts looking like an old biome. So what does it matter? You have all these bacteria, you cause a stroke, the gut biome ages. So we decided to do a fecal transplant where we took young biome and put it into old animals, and old animals got young biome. Our young, 5 minutes, 2 minutes, I'll go even quicker. All right, so the young biome was very protective. And in fact, we were able to improve cognitive outcomes and depressive phenotypes in old animals that got young biome. The young animals, though, they got the H biome, they all died. It's amazing, like if there's something super toxic. So we didn't know if it was the bacteria or something the bacteria produced. They produce a lot of these short-chain fatty acids, and again, it kind of ties in nicely with Dr. Sanson's talk, because these are very involved in metabolism. So what we decided to do, because it's kind of yucky to think about fecal transplants, right? Poop transplants. So we turned out, what we decided to do is genetically engineer some bacteria to overproduce some of these fatty acids that we found were very beneficial. So basically, we overproduced 5 bacteria, gave them a thing called inulin, which is what they use to make fatty acids. We gave a stroke, and importantly, we waited 3 days after stroke. We're talking stroke recovery. We can't predict when people are going to have strokes, right? So we can't change the biome before the stroke. So we decided we're going to change the biome after the stroke. And what we found is if we gave this cocktail of short-chain fatty acid producers at day 3 and 4 after stroke, they had tremendous improvement in their neurological deficit scores. They looked almost normal. And the gut looked almost normal. So this was a vehicle, and this is with these short-chain fatty acids. See how nice and plump the gut looks here, very healthy. This is all deteriorated. This is mucus. You can see the mucus barrier is completely absent, and now it's completely restored just by giving these 5 bacteria. We could see that those short-chain fatty acids were higher in the gut, they were higher in the plasma, and they were higher in the brain. So all we did was give 5 bacteria to the gut, and we found an increase in brain fatty acids. The cecum was much less distended. You can see here, this is a normal, healthy-looking cecum. And then finally, we checked, well, does this have any relevance in patients? So this is age-healthy controls and stroke patients. And you can see what happens after stroke. This is 24 hours after stroke in patient serum. You can see it's like black and white. Well, it's actually yellow and blue, but you can see this age-healthy individuals look completely different in their short-chain fatty acid profile than patients with stroke. Their short-chain fatty acids just completely are depleted. And this is probably important to recovery. All right, so final conclusions. Women are disproportionately affected by stroke. Women are older at the time before stroke. Importantly, they may also be ineligible for trials, because many of those clot retrieval trials where you suck out the clot, they said you have to have a thing called a modified Rankin of 0 to 2. Women, if you're using a cane, your modified Rankin is 3. You may not even get the opportunity to be in a trial. So that's an issue we have to know. If you look at endovascular stroke therapy, this is not out yet, but it's in press. We looked at people who got that clot retrieval, and women actually gained more healthy years of life than men. Stroke induces a profound disruption of the gut barrier. Aging leads to high baseline inflammation that can be reversed. We've also done it with bone marrow, not just the fecal transplants. We've given aged animals young bone marrow, which replaces the youthful immune system. And we also do a thing called parabiosis, which is when you sew a young mouse to an old mouse. So we need to recapitulate. Yeah, it's cool though. I can talk about it later. I am aware people hate me, but it's, so basically within 8 weeks, they completely share blood supply and it rejuvenates the aged. Unfortunately, it also ages the young. And it is much easier to age a young animal than to make an old animal young, unfortunately. Basic science should recapitulate these findings. And that's it. And this is my laboratory. It's funded by the American Heart Association and the NIH. Thank you.
We have time for maybe 1 or 2 questions. So I'll ask a quick question if no one else has one.
No questions.
So Claire, yeah, go ahead.
It's not so much a question, but I would really like to thank you because your definition of 3 different kinds of strokes was very articulate and really was meaningful to me because my father passed away from a stroke.
Right.
And if you do not educate the public, which I'm thankful that you're here to do today, people will not know how to react and what kind of questions to ask. And I'd like to just thank you for that.
You're welcome. And it's important because that clot busting drug, and I didn't show the data, we actually have a mobile stroke unit. We treat patients in the field. We have an ambulance that goes and gets the patients that has a CAT scan in it, and we will treat them with the clot buster right in the field. The problem with delaying this is every minute you lose a lot of neurons and your chances of recovery go down. But you have to get that CAT scan because you never would want to give that clot buster to somebody with a hemorrhage. They bleed, they bleed more and they would die. So we are now able to find that out in the ambulance and then give the clot buster, and that saves us probably like 20 to 30 minutes, which is important. But it's important to know the different types of stroke and how you treat them.
So my quick question was, do you think you need all of these short chain fatty acids to achieve putative effects, or can one of the short, like if you simply take butyrate?
So butyrate is the big one, and I know you love butyrate.
Yeah.
He loves butyrate. Yes, he's a butyrate fan. The thing about butyrate is, and all these short chain fatty acids, is it's probably not going to work to give the short chain fatty acids themselves. And they say, why did you make bacteria that make them? Because a lot of times they're not going to get through the gut. They're going to get destroyed in the acid. You have to get constant infusion. So if you can instead restore bacteria that lives in that niche and produces it continuously over time, that's probably going to be a better approach than trying to continuously give somebody oral butyrate, because it's probably not going to work. We have tried to give catheter-based butyrate directly to the gut, and it does work. But it makes a lot more sense to have the bacteria because then it's not just a one-time deal. You can do it for days, months, whatever.
And just the second quick question is are the concentrations that you actually reach in the brain adequate to affect different targets that are, because you often need high micromolar or millimolar concentrations, which might be very difficult.
But we're getting there. So with the metabolomics, it was amazing to me. I did not think that we would see a change in short chain fatty acids in the brain giving gut short chain, but we did. And we found those downstream effects were activated. The receptors were activated. So.
Fantastic. Thank you. All right, so our next and final speaker of the morning is R.G. Hillis, who's the Executive Vice Chair of the Department of Neurology at Johns Hopkins. And she's also, she has many titles, the Director of the Stroke Division, and the Center of Excellence in Stroke Detection and Treatment. I think in heading the stroke program at Hopkins, They recently got a big shot in the arm from a $50 million donation from the UAE. And so I think it's going to offer real opportunities for some really transformative experiments, particularly in human. But she's best known for her work on language, and she actually has a very interesting background, and then she started out as a speech and language pathologist, and then went to medical school at Hopkins, did a residency there, and has really catalyzed really important natural history understanding of language problems after stroke, how they evolve using imaging and very detailed outcome measures. And she's even been able to identify important therapeutic targets for improving language in the acute setting. She was here visiting us in January, and I can tell you you're all in for a treat. Welcome.
Well, thank you so much, Raj. It's really an honor to be back here. I had a wonderful trip here before and met with fantastic neuroscientists doing wonderful work. And so I'm just thrilled to be back. And I'm going to switch gears a little. I'm going to be talking about problems in speaking, discourse, narrative speech after both left and right hemisphere stroke. So most people think that language problems only occur after left hemisphere stroke, but different types of communication problems occur after right hemisphere stroke, and I'll be discussing those. And then show that these problems do improve over time, and describe some of the predictors of recovery and interventions that augment recovery, and describe deficits in recognizing and expressing emotions after right hemisphere stroke and how those recover. So this is a picture known to all stroke neurologists. It's actually originally from the Boston Diagnostic Aphasia Exam, but it's a picture that's used as part of the NIH stroke scale. And we only use it for the NIH stroke scale to say, well, the person has aphasia, they don't. But you can quantify the number of content units that the person produces. So there's actually a normative set of content units, not actual words, but ideas. So something like disaster or chaos or something would be the same content unit. Mama, mom, woman would be the same content unit. So there is this set of norms, and then we characterized, me and other people have characterized the number of content units that normal people express. How many of those does a patient produce after a stroke? And also the number of syllables per content unit, which is a measure of the efficiency of communication. And I'll show you an example of that. And after left hemisphere stroke, people do get the main idea of the picture, but they have difficulty expressing it. They often are telegraphic, just produce content in very short sentences or single words, and they have difficulty with sentence structure, or they're very fluent and they produce a lot of jargon, or they're just anomic, so it takes them a long time to get out their ideas. So they say, well, the woman is, she's doing the thing that you do with one description, for example, from a left hemisphere stroke patient was a toy, no, boy and girl, oh no, oh, wham, ouch, geez, naughty, woman, wash, disaster. And so they get the main ideas that people say, boy, girl, woman, washing dishes, water overflowing in the sink, she's not going to pay attention. They get the main ideas. People with right hemisphere stroke say things that are true, but often not what normal people say. So this is one of my patients who said, Well, it's on 8.5 by 11-inch paper. Overall covered by plastic. Looks like it may have been done with drawing pens and India ink on white paper. It's less than 20 pounds of paper. I was just going to use black to keep it from shining through. And then I redirect and say, tell me what's happening in the picture. He says, well, I see a window and curtain. Someone has pulled back, and hospital-type curtains exposing a window and utensils on the table, a pan or a pot. Curtains tied back with strings tied, kitchen curtains, no particular design. The rest of the curtains only called curtains because of their overall lack of color. There's evidence that the paper was pushed out for 3-ring binder. The room seems to be filled with air because the curtains have a billowing effect. Now, all of these are kind of accurate, but normal people don't say any of them. It's very few actual content units. The curtains are a content unit, but you can see it also shows he has left neglect. That is, he's not paying any attention to the left side of the paper. It doesn't mention the boy or the girl or the jar. So we looked at the number of content units on average of all comers of left hemisphere stroke, right hemisphere stroke, and controls, and you can see there was really no difference between the left and right hemisphere stroke in getting accurate content out, but they were both significantly different from controls. And syllables per content unit, that is, the efficiency was impaired in both left and right hemisphere stroke compared to controls. And then the ratio of left to right content units, this is a measure of neglect again. People who have a right hemisphere stroke neglect the left side of the picture, and people who have a left hemisphere stroke often neglect the right side of the picture. And so the ratio of left to right content units can be quantified. So with this 1-minute test, and it literally only takes 1 minute, you can find out a lot about the stroke patients. And in fact, we found that, so this just shows the statistics from those bar graphs, but we found that total content unit negatively correlated with lesion volume for both left and right hemisphere stroke. So you can get an idea of the lesion volume just with this 1-minute test. Total content unit actually correlated with percent damage to specific regions. I'm not going to go into this. Also, syllables per content unit significantly correlated with percent damage to specific regions in both left and right hemisphere. And the ratio of left to right content units also correlated with the percent damage to particular regions. So with this 1-minute test, we can get an idea of not only the volume, but maybe where the stroke is. My lab also looked at whether you could predict first of all, does this narrative recover? And what are the predictors of improvement in the number of content units? And this is Lindsay Keeter from my lab, who found that age, and this goes back to what Louise was saying in mice and the people, education, baseline content units, severity, as well as full lesion volume and percent damage to the infarcted temporal lobe were all significant predictors in a multivariable regression analysis. Now there's improvements that improve narrative speech, and I'm not going to talk just yet about language therapy, but in fact there's some acute interventions that Louise talked about a lot, but there's also sometimes real challenges. You can't do embolectomy or the clot buster in everybody. So we recently saw a 76-year-old woman who had had a left carotid endarterectomy 3 times, and she then had recurrent severe narrowing of the left carotid stroke or left common carotid, and had some small left hemisphere strokes. So then she had stenting because they couldn't do the recurrent endarterectomy again. So she had a stenting of the left carotid, and that opened it up for a little while, but then it became occluded also. And once it's completely clogged, you can't do anything about it. So she had some fluctuating symptoms, including transient aphasia and right arm weakness. But it improved lying down. And she could name pictures, or she could name stethoscope and nouns and things, but she had severely impaired sentence production or connected speech. And opening the occluded Stent was not an option, but she also had 70% stenosis of the opposite carotid artery. And so we thought maybe in doing a stent of the right carotid might get more blood flow to the left hemisphere. And it was worth a try. And following the stent, she improved in content units significantly, as well as the total number of syllables and the syllables per content unit. However, she really only maintained these gains when she had moderately high mean arterial pressure. And we know how important it is to control blood pressure, particularly in midlife, and to continue to keep it low. But in acute stroke, sometimes you actually need a little bit higher blood pressure to get blood flow to the brain. And in her case, we did. Sometimes we could get blood flow to the brain with her just lying down. She get more music sitting up. So we gave her a medication to kind of increase her blood pressure to a moderate level, not to 120 over 80, which is our goal, but more 150 over 90. And in rehab, they thought, no, we know stroke patients are supposed to have low blood pressure, so they stopped the medication. She was readmitted, and her MRI scan showed that she had, again, extended her left middle cerebral artery stroke. She again had significant decline in both syllables, in picture description, and content units. And so we gave her fluid and Metadrin, and she showed significant improvement in her narrative speech measures. And then she was maintained on Metadrin, just keeping the mean arterial pressure over 100, which is, again, moderate. It used to be considered normal, but now is considered a little high. And so this just shows you she had, this is, this is her occluded stent on the left, and radiology left is always on the right. This is her left hemisphere that has very poor blood flow. She had a right hemisphere stent placed, and now that is very severe blood flow reduction. Green is less severe blood flow reduction. And still she only has this tiny left hemisphere stroke, very little. But she was discharged, her blood pressure dropped, she became a little dehydrated, she came back in with this new stroke, and again had, this is after she had some fluids, she still has a poorly perfused hemisphere. So we looked at these measures I was talking about with narrative speech, and you can see that she improved, and this is content units, after the stenting. And these all 20 is probably, the normal score is about 18. So she improved from almost 0 to about half normal. Her blood pressure dropped, she went down. When she came back in, gave her fluid, she improved again to almost normal. And the same thing, looking at syllables, this again in green is normal. She improved a lot with the stent, but then got worse when her blood pressure dropped and back up. And the same thing with syllables for content unit. Green is normal, they're low. She started out very poor and improved with each of the interventions. So then we asked, what predicts recovery of naming, the most common deficit in after left hemisphere stroke? And we did a longitudinal study of naming recovery. So we evaluated patients at 0 to 3, 6, 12 months, and we found that lesions in the lesions in the superior temporal gyrus or the arcuate fasciculus. Superior temporal gyrus is known to be important for language, and the arcuate fasciculus is a white matter tract that connects some of the areas important for language. Really predicted, the percent damage to these areas predicted how well somebody recovered. If they had more damage to these areas, they recovered naming less well. And then we looked at a very large cross-sectional study of naming outcome in chronic stroke patients, and areas in red. Remember, everybody in this study had strokes. People had strokes in the red areas failed to improve in naming, but people who had lesions in the yellow areas actually show good recovery of naming. These are less important for naming. So these look very similar to the areas we found in a completely different subset of our population of patients. We also found that we looked at other variables, and one of the variables Karen actually talked about is white matter disease, or sometimes called leukoariosis. You can see that all this white stuff around here, which is really a marker of kind of bad brain. It's caused by diabetes and hypertension, as well as other things. But we evaluated the independent effects of right hemisphere leukoariosis. We didn't use left hemisphere just because you get white matter changes from the stroke itself. And age, diabetes, hypertension, and damage to particular areas of the brain could predict recovery of naming. And the ones that were most important were damage to the left superior temporal gyrus, which I just showed you in a separate population, the number of months post-onset, because people keep getting better over time, and the severity of right hemisphere leukoariosis or white matter disease together predicted the outcome of naming. And the only ones that were independently significant were this superior temporal gyrus lesion and right hemisphere leukoariosis, which both predicted poor recovery. We found the same thing looking at a different measure of narrative speech, word fluency, and we found looking at all of these variables that the only variables that were significantly associated with recovery were right hemisphere leukoariosis and damage to the left superior temporal gyrus. So this is just an example of a patient who has a big left MCA stroke, but has a very healthy looking right hemisphere. And this patient recovered naming very well, despite this huge stroke. This patient had a tiny left thalamic stroke, which can cause aphasia, but had a terrible looking right hemisphere and the rest of the left hemisphere. So they just didn't have a lot of brain to sort of reorganize and to take over the functions of the damaged part and failed to show good recovery. Treatments that seem to improve outcome are language therapy if given in adequate doses. We are currently doing a study of language therapy plus transcranial direct current stimulation, which is a very low amplitude, non-painful, non-invasive stimulation in acute stroke. And I don't know the results of that, but there was a recent clinical trial. It was a sham-controlled trial of 72 patients published by Friedrichs and al. In JAMA Neurology recently that showed a significant effect of transcranial direct current stimulation when given with language therapy compared to sham with the same language therapy. They showed a 70% greater improvement in naming words that were not practiced. The other thing that was interesting is that the genotype mattered, that patients who had a particular genotype in actually the BDNF gene showed greater response to aphasia treatment than those who had a MET allele, either MET-MET or McDowell, regardless of whether or not they got the TDCS or sham. So we're looking at that in our trial as well to see if that is true in acute recovery. And other interventions we've used to improve blood flow soon after stroke. We've heard about thrombolysis and vascular therapy. I mentioned temporary blood pressure elevation. We don't want to keep the blood pressure up for very long. But in acute stroke, where there's an area of low blood flow beyond the infarct, there's a loss of autoregulation, so that increasing the regional or increase in the mean arterial blood pressure actually increases the regional cerebral blood flow in that area. And these are just some examples. This is a patient who had very poor naming and had good comprehension and had poor perfusion, shown in blue, of this left temporal area, and recovered when we restored blood flow to this area, showing recovery of naming. And this is a similar patient who we just improved her naming, both oral and written naming, in 1 day, just by increasing the mean arterial pressure from 80 to 100 temporarily, and then we normalized it. And this just shows the relationship in acute stroke, that if you increase the mean arterial blood pressure, the naming can improve in a patient who has this big area of hypoperfusion beyond the infarct. The person had a tiny subcortical stroke. So it was really just dysfunctional tissue. So even out to 4 or 5 days, by restoring blood flow to that area, we could show improvement in naming. Recent trials keep extending the window that we can do something like pull out a clot. Now it may be 24 hours, it may even be longer according to these data, that if you can restore blood flow in somebody who has an area of low blood flow beyond the infarct, it may be helpful days out. And this is just one more study showing the same thing. Well, what about the right hemisphere communication deficits? I talked about narrative speech being abnormal, but the other thing that's often abnormal is the recognition and expression of emotion through voice. So changes in pitch, rate, loudness, stress, and rhythm. You all know that it's sometimes not what you said, but how you said it that conveys meaning. And it's really crucial for communication, interactions, and well-being of speakers. So if I say to you, that's a pretty dress, you may not think that it was a compliment. But likewise, what we often see is patients who say, I had a wonderful time. You don't know if they mean it or they don't mean it, because they don't sound like they do. So the right hemisphere stroke patients are often thought to be apathetic, they don't care about others, they don't care about their own deficits. So this is called affective paucity, and both comprehension and expression are more impaired in people with right hemisphere stroke compared to controls or people with left hemisphere stroke. However, propositional paucity, which is like stress on that conveys meaning like affect versus affect, can be impaired after both right and left hemisphere stroke. So some right hemisphere strokes impair recognition of emotional prosody, and this is one of my postdocs who studied this, and she found that mean accuracy or comprehension of a person's emotion after right hemisphere stroke was only about 50%, whereas controls, age-matched controls, were at least 83%, and identifying if a neutral sentence like we had hamburgers for supper was spoken with an angry tone of voice, we had hamburgers for supper, or a happy tone of voice, we had hamburgers for supper. So they were, we found individual predictors of emotional recognition were older age and greater damage to the right posterior superior temporal gyrus. So again, it's a very important area of the brain on both the left and the right. And it was for most emotions, or all emotions were significantly different for controls. This is the area that seems to be important. And then the only area that was important for a specific emotion was the amygdala. So damage to the right amygdala was associated with impaired recognition of fearful prosody. And that has been shown in many studies that the amygdala is very important for recognizing fear in both facial expression as well as we found voice. Other right hemisphere strokes are associated with impaired prosody expression. And this is a young faculty member in my lab, and carried out this study. And we looked at the percentage of voxels that were damaged in specific areas that correlated with these acoustic features. And we used these acoustic features because they correlated with listener judgments of poor prosody or flat prosody. So it was spectral flatness and coefficient of variation, or that the variability in the fundamental frequency or pitch of the voice. And then we tried to find out if there were specific areas of damage that were associated with abnormalities in each of these acoustic features independently of age and sex. And we found, we looked at 9 different regions, and together sex, lesion volume, and damage to specific areas, mostly in the frontal cortex as opposed to the temporal, and white matter tracts were associated with poor expression of prosody through changes in pitch. We found basically the same thing with our other measure of prosody expression. So impaired emotional expression after right hemisphere stroke seems to be due to changes in specific acoustic features caused by infarcts or strokes in right inferior frontal gyrus. The supramarginal gyrus is part of the parietal lobe or associated white matter tracts. So what can we do about that? Well, it turns out some people have some people have flat prosody because they are impaired in knowing what emotions normally sound like. They've just forgotten what angry should sound like. And we identified lesions associated with this deficit and determined if it can be improved by giving them clues. That people sound sad, they usually have slow speech, low pitch, monotone, quiet, as opposed to angry, which tends to be loud, fast, and so on. And so we found 8 right hemisphere stroke patients who had reduced pitch range compared to controls. And we found that 6 out of 8 improved to a normal range when we gave them these kind of cues, when we told them what they should be sounding like. The others probably had impaired expression due to a more motor deficit. So they improved significantly with these cues, whereas healthy controls actually show poor pitch range with the same cues. They seem to be trying to force a change in voice. So recognition of affective prosody is frequently impaired after right hemisphere stroke, and distinct areas of the right hemisphere are associated with recognition versus production deficits, and deficits improve with treatment directed to specific underlying problems. So it's important really to educate family members of people with right hemisphere stroke that patients may have difficulty with emotional prosody, perception, and production, and to tell them to explicitly state their feelings. So it's good to say, I'm angry with you, or I'm really happy about that, just rather than trying to express it with tone of voice. And that can prevent some frustrating communication, and then treatment directed to the underlying cause may be helpful. So this is my last slide. Both left and right hemisphere strokes can cause deficits in communication, but the deficits depend on the location of stroke as well as the size of the stroke. They generally improve over time, depending on percent damage to a few critical areas and their white matter tracts, as well as age, education, the health of the brain unaffected by this stroke, and appropriate treatment. And I'll just thank the people who do this work in my lab, in the cerebrovascular division, and funding from NIH, and some of my colleagues who've done the work. Thank you very much.
Time for one question.
What's the name of the horse?
I'm curious if you can maybe speak to differences in deficits or recovery that you see in bilingual patients.
So that's a great question, and it doesn't have a simple answer. So some people improve in all languages together, and some people improve in a single language that you treat, that's less common. And some people after stroke are more impaired in their mother tongue versus other people are more impaired in the language they use most commonly now. So it's very variable across patients.
Great. Thank you very much, Harry.
So, next, item item on the agenda is to give out our annual McDowell Awards. And we decided that in addition to having the symposium, that it was really important to create a mechanism by which we could celebrate individuals who given importantly either to the Institute or to neurorestoration generally. And our first McDowell Award, maybe first I'll ask Lynn and Laura to come up and stand with me as they're going to be giving out these awards as I announce the different. So our first McDowell awardee is Dr. Bruce Dobkin. He's currently the American Professor of Neurology at the David Geffen School of Medicine at UCLA. Following in the footsteps of Dr. McDowell, it's widely, I think, believed that Bruce is one of the most, if not the most, well-known neurorehabilitationists in the world. Last night we were joking at dinner that he is the Elvis Presley of neurorehabilitation. He began his tenure at UCLA as the director of the stroke program, so very fitting that this particular year he would be honored. He then created and directed the first neurological rehabilitation and research program, which included an inpatient service, an outpatient service, and most significantly, repair-related research. And I think it really reflected an extension of things that we were trying to accomplish here at the Burke Neurological Institute many years ago. He's been funded by the NIH and foundations for a long, long time. I think maybe since those organizations were founded for many different studies for understanding movement after stroke, for functional imaging, clinical trials, and as principal investigator of a training grant, again, an important part of his legacy for postdoctoral fellows in restoration research. He's participated in numerous important efforts that have happened locally, nationally, and internationally, including the NINDS Pilot Stroke Data Bank, the first external carotid, internal carotid bypass trial, the Tycobi trials for treatment of transient ischemic attacks and secondary stroke prevention, hemodilution, and other acute stroke trials. Bruce began studies of body weight supported treadmill training in 1990. These were really, I think, the first trials that were done that used robotics in some way to think about how we repair the nervous system. And that was published it actually led to a NIH-funded multi-center spinal cord injury locomotor trial, which was published in Neurology in 2006, and then a following study, which was actually published in the New England Journal of Medicine, the LEACH trial. He has done important work on studies of feedback to improve walking outcomes. He has contributed to the design and served on safety committees of the EXCITE trial, the only positive trial for repair, the VA upper extremity robotics trial involving the robots that actually were developed with MIT here at Burke, and half a dozen other randomized trials. In collaboration with UCLA engineers, he's developed these really nifty activity monitors that allow us to query what patients are doing actually when they go home, because it's not what they do when they come in for their therapies, but what they do when they go home that often really matters. It's a long list of accomplishments, as you can see. He was the founding editor of Neurorehabilitation and Neurorepair, which is, again, widely considered the leading journal in the repair field. And while all these accomplishments, and I haven't even begun to touch the iceberg of, the tip of the iceberg of all the things that he's done, they do not begin to tell the story of Bruce's unique spirit and vision. And last night we had a chance to hear a little bit about this. I think one of the other reasons we wanted to honor him is in 2005, he was approached by he took care of a very wealthy patient who wanted to show his actual gratitude, and Bruce could have very easily just taken all the resources for himself. Instead, what he did was to create a unique foundation called the Adelson Foundation, which brings together individuals who study cells and animal models and do human clinical research together twice a year to bring together a unique collaborative model of how we can advance our understanding of repair-related treatments that are not defined by a single disease and that actually might affect many diseases. And this model has been remarkably successful now in its 13th year of really breaking down silos, of bringing people with really different core competencies together to try to tackle transformative problems. And Bruce deserves enormous credit for that. I think, as we were talking about last night, there are now 104 investigators that are sponsored by this foundation at a support of nearly $60 million a year. So if you just do the calculations times 10 and 13, that's quite a bit of information that has been able to be generated related to repair. So I think another thing that's very exciting that has come out of that is the recent discovery of a drug and a target that can actually potentially help people recover from stroke in the chronic phase, a drug called Moravro. And he's working with Burke investigators and with Lauren from Yale to really think about how we are moving that forward into patients. So really for your remarkable visionary efforts in advancing and solidifying neurorestoration as a bona fide clinical field, and your unselfish, creative, solution-driven impulses that led to the formation of the Adelson Foundation, we honor you with the 2019 McDowell Award. And before you come up, Bruce, one of your maybe your most famous mentee, Tom Carmichael, told me that I could give you one of these koosh balls because it reflects a story of how your constant resourcefulness is being used to try to improve rehabilitation. And maybe I'll let you take this, tell that story as you come up and accept your award.
Got this one. Thank you. By the way, I knew Fletcher really well. Spent lots of hours with him. He's always just a wonderful man and just a gentleman and an inspiring man. And this is very special.
Thank you.
Need to be his own. Congratulations.
Yeah. Well, the story, I'll tell it because Bruce didn't, was that as his kids started not needing the use of these koosh balls anymore to play at home, he brought them into the neuro rehab unit, and they found that it was in many ways more effective than any robot. that they had ever developed in getting patients to be active and excited about their physical therapy.
You get a kick out of this. They all had personalities and names. And it became a kind of, it was like having a pet dog. But it was a lot easier to keep.
So our next McDowell awardee is Robert Baldoni, Chairman of the Board of Trustees for the Burke Neurological Institute. I think, as I was mentioning yesterday, I think when Don Foley, who was then chairman of the board of the Burke Hospital, tapped on Bob to be chairman of the Burke Neurological Institute nearly a decade ago, Bob was probably telling himself that he didn't run fast enough. But I think that in talking to Don, it was a very carefully selected decision, because we needed somebody who not only could potentially throw the ball for us, but could be kind of like a blocker when we needed him. And as you'll see, Bob is ideally suited intellectually and physically in both of those roles. The Institute at that time was still in an uncertain phase of growth, and the prospect of trying to provide fiduciary oversight to a bunch of scientists sounded more like herding cats than gently steering the rudder of the ship. The challenge was compounded by the fact that on September 29th, 2008, as many of you remember, the stock market fell nearly 800 points, causing this institution and others to rethink their growth strategies. Indeed, we had just brought on 6 new faculty, many of whom you'll meet at lunch. We now have brought on an additional 8 new faculty, so a total of 14. And I had made commitments to them that we would be renovating new space, and the board was beginning to rethink whether that was actually a good idea. And while many on the board were uncertain about what we should do, Bob, working closely with Don, actually created a path for immediate strategic and financial analysis that argued, in fact, that we should go forward with the renovation despite the large hit our endowment had suffered. The end result was that the faculty were hired. I could maintain my commitments. They were placed in new space that they were promised, and the table for remarkable growth in the Institute was set. During the past 10 or more years, we've grown from just over 50 employees to more than 110, and an NIH budget which would be in the top 25 of neurology departments, but would be #1 compared to PM&R departments in the country. In achieving this remarkable growth, Bob, your leadership has shown through in many ways. First, in the carefully considered and organized way you consider and advise on large and small problems in the Institute, from choosing a rebranded branding company to reading over legal documents for our Burke collaboration, to selecting new members of our board. Your willingness to roll up your sleeves to do anything requested of you has been remarkable. Second, you have been the single most generous and consistent board level donor to BNI over the past decade, and this support has allowed investigators to seed risky projects that may have never had the opportunity to see the light of day. It has been marvelous to see through the depth and engagement of your actions how much of a stalwart supporter and advocate you are for what BNI is trying to accomplish in curing chronic neurological disability. In addition to sharing your remarkable talents, we've also had the benefit of having your son Robert work in the Psychology lab, and for a number of summers he's at the Institute. I think we would all agree, I can think of no greater testimony to your faith in us. BNI is on the cusp of truly transformative initiatives and discoveries, and much of this ascendance can be traced to your support and oversight. We're proud of that. All right. Well, we expected long speeches, but maybe this is our trajectories. So, last and clearly not least, all right. So it's a great pleasure for me to introduce the last McDowell Award winner, Dr. Frederick Maxfield, who's the Vladimir Horowitz and Wanda Tostanini Horowitz Distinguished Professor in Neuroscience and Biochemistry and Chairman of the Department of Biochemistry, as well as Acting Chairman of the Department of Cell and Developmental Biology at Weill Medical College. Since the late 70s, you and members of your laboratory have pioneered exciting new mechanisms for allowing biologists and clinicians to peer into the inner workings of cells inside and outside of the nervous system. Your detailed investigations have helped foster the development of fluorescence imaging microscopy, a discipline which permits us to use low levels of probe-emitted fluorescence quantitatively in living cells and are now well documented in cell biology textbooks around the globe. The focus of these efforts has been on understanding how cells constantly communicate and monitor their environment, how cells in our bodies eat debris and toxic proteins, and how cells homeostase essential lipids such as cholesterol. Your studies have had direct implications for diseases of interest here at VNI, including atherosclerosis, stroke, Alzheimer's disease, and ataxias. In accomplishing these remarkable scientific feats, you have trained dozens of graduate students and fellows and faculty and built one of the leading departments of biochemistry in the world. As remarkable as these accomplishments are, they only begin to tell the story of your unique talents and how they've contributed to BNI and other organizations like us. Your wisdom goes beyond the scientific method to an uncanny sixth sense style of clear thinking. Your ability to see a logical and decisive path forward where most of us see darkness or fog has constantly astounded and delighted us. As part of our scientific board of directors, you've donated your time with incredible diligence and attention to provide remarkable, thoughtful wisdom. In helping us to chart a path forward, you've shown us how to disagree without being disagreeable. You've to challenge without undermining the confidence of those you challenge, and to constructively criticize in a way that builds and grows rather than decays. You are kind but not wishy-washy. The ascendance of the Institute, while incomplete and ongoing, could not have happened without you. Having had the immense pleasure of knowing you for more than 35 years, and having directly benefited from your mentorship during that time, it's really an honor.
Well, Raj, I just want to, partly I understand why people sit down quickly after your introduction. I sort of feel like doing that myself. But I would like to say a few things. First, I have been at Cornell since 1995. I regret to say that I never really seriously interacted with Dr. McDowell, and I really wish that I had. I have interacted with many people at the Burke since even before Raj came, Gary Gibson, John Glass, among others. And it's always been a pleasure. And so Raj mentioned that I helped with training him as a MD PhD student. And of course, this is one of the greatest things, the greatest honors you get is to train young, bright people and then watch them flourish.
Oh.
I have to say, I was delighted when I learned that Raj was going to be coming here to become the head of the Burke Medical Research Institute, now the Burke Neurological Institute, because I knew that he would do a terrific job, but I didn't really know how terrific. And I think one of the things that we haven't really talked about is Raj has directed this institute to take on what I think is one of the most challenging problems in biomedicine. Now, we heard this morning about things that happen to prevent stroke, things that you can do in the first few days. Raj is really directing the institute to work on chronic neurological disability. That is, people who recover maybe for 30 or 60 days, maybe a few months, and then it often plateaus. And Raj has decided that that is a problem we ought to be able to attack. And he's really directed the Institute towards that. And I just can't say enough about how exciting that is and how glad I am to see you doing it.
Thanks so much, Phil. Fred didn't tell the full story, which I'll finish with, and then you get to lunch. And that is that I had a wonderful thesis advisor, but the initial project for my PhD and the initial project they put me on was one that was slated to fail, and so I was working with the spectrophotometer, and Fred, in his very gentle way, said, "I need to talk to you in my office." I said, "This project isn't going to work, and we need to figure out a way of gently telling Michael, who again was a wonderful advisor, that you need to get on to a different project." And so he not only, and so my advisor was the chair of the department, and he was a relatively new assistant professor at that time, just indicating how old we actually are. He arranged it so that I could actually do a project that not only was doable, but that was much more transformative. And Fred turned out to be one of my co-mentors, and we were able to publish a lot, but I would probably be still doing my PhD. And with that, we'll have a round of applause both for Lynn and Laura. And
I did get to ask.
Yeah, no, that is.
A couple of questions I want to ask you.
And she said she was going to your brother said
It's much better to have it sort
So this is next one.
Yeah. I'll do that. But I've learned, I'll never do
So,
oh, well. I thought it was
coming in. So I think before I start, I just wanted to take 2 minutes to tell you about an exciting initiative that we're beginning in the Institute. In February, we were fortunate enough to receive $5 million from the Empire State Development Corporation to develop a clinical trials network for chronic stroke for patients who have upper extremity disability and stroke. And the idea of the platform is to actually set up a trial structure that represents our long-term commitment to try to cure chronic neurological disability in stroke first, and then extending it to many diseases. And the platform will involve a standard 6 weeks of robotics training, which will not only standardize experience for patients, but also represent quantitative outcomes. I think the concept here is that fixing the brain is not like fixing the kidney and the liver, that you need to fix the hardware and somehow shape that hardware with experience. And so a team of individuals here in the Institute over the past 2 years are working with the state to develop a business plan and the affiliation agreements. And the effort involves 3 institutions: the University of Rochester, the Wadsworth Laboratories at the New York State Department of Health, and the Burke Neurological Institute, working closely with Dan Hanley and the BIOS at Hopkins and the Trial Innovation Network. And the great thing about having the BIOS is that Steve Mayo is a part of that, and he's run a CRO named Emissary for 30 years. So the goal is to create sort of the openness and transparency and maybe the sustainability of academia, which will allow us ultimately to be testing combinations of drugs, biologics, stem cells, or electroceuticals in combination with standardized training, and up to 4 to 6 interventions a year. We're imagining that there will be failure on the path to success, but that this is a strategy for testing in humans, whether combining experience with something that unlocks the plasticity of that experience will be useful. So if you have any questions about that, we can answer them afterwards. It's a great pleasure to introduce our next speaker, and somebody else who I benefited a tremendous amount from when I was training. She was actually my senior resident, Becky Eichhorn. She's the director of the pediatric stroke program at the Perelman School of Medicine at the University of Pennsylvania. And Becky, I think we were talking about this last night. They're great professors in Hopkins neurology, but much of our learning came from our fellow residents, and I was so fortunate to benefit from Becky's presence. In addition to being a great teacher, Becky has built a world-class program in pediatric stroke. We don't usually think of stroke affecting kids, but as you'll hear about, it definitely does, and I think it's opening doors for really understanding prevention, treatment, and repair in this population. So thanks very much.
Thank you very much. I guess you can all hear me. Thank you so much, Raj, for bringing me up here to this really inspirational meeting. And my being here reminds me that my gestation and birth as a scientist in neurology really began at Hopkins. And that inspiration started with interactions with brilliant co-trainees such as yourself, such as Louise, and other people who I've met along the way. So I feel like I began at Hopkins, and it seems sort of fitting that my connections then are now helping me to be here and to continue to be inspired. Your work and that of everyone at this place is really inspirational. It's kind of giving me new a new way of thinking and some new energy. So I'm going to talk about really the clinical aspects of childhood stroke, and since most of you probably haven't had experience with any childhood stroke, it often comes as a surprise, and it's frequent when I give a presentation and the first words out of people's mouths are, "Oh my God, I didn't realize children could have a stroke," but absolutely that is the case, and Louise already alluded to that fact. So these are my disclosures, nothing really important. And this gives you an idea of where childhood stroke research is, and it's really in a booming industry. At the same time, we're still, I think still in our toddlerhood compared to where research has gone for adult stroke and adult brain disease. And that's both a good and a bad thing. It's a good thing because it really energizes a lot of people to realize there's so much to learn and there's so much to contribute that even a small contribution can make a big difference. And that's the way I have felt for these last 15 or so years that I've had the privilege of working in this area with equally imaginative and inspired people in childhood stroke. And we're small enough still that we consider ourselves a kind of a small family. And when we have our investigator meetings, we welcome new people to the group, but we have a long history of knowing each other as individuals, and that's one of the great pleasures of being in this field. And it's true of the people in pediatric stroke, but it's also true of a pediatric stroke expert within the larger community of adult stroke. And again, my hat is off to the adult stroke people, particularly the women who have inspired me and helped me to feel as though I belong and can make a contribution. So just as you heard, there's a spectrum of different types of stroke in adults. There is similarly that in childhood. And certainly children, it doesn't work out like this. Children can experience an arterial ischemic stroke, and that's the type of stroke most people think about when they think of a child having a stroke. But in fact, children can also have a TIA, which precedes their stroke. But importantly, children can have a hemorrhage. The causes of stroke in all of these subtypes is very different in children. And one thing that I hope to take away from my trip here is a better understanding of the underlying biology in hemorrhagic stroke. In contrast to adults, hemorrhagic stroke makes up about 40% of stroke in childhood. And so this is a really big area of need and of opportunity. In addition, children are prone to have venous thrombosis, which can also lead to lifelong disability. So those of us in this in childhood stroke really have to understand and deal with all of these subtypes of stroke. And yet there's another subtype, and that is children who have stroke at or around the time of birth. And this is yet very different in terms of its causes, its manifestations. And just as in childhood stroke, there are ischemic subtypes, which can present acutely in childhood with, or at the neonatal period with seizures. There is neonatal venous thrombosis, and then there are hemorrhagic lesions. And hemorrhages in the neonatal period are also about 40% of all perinatal stroke. So we have a lot to learn and a lot of opportunities and need to understand that. And then there are children who had a stroke but was silent in the early period after birth, and it presents itself as an infantile hemiplegia. And this is one type of cerebral palsy, which has been the subject of fantastic groundbreaking work by friends and colleagues of yours at Blythedale. But this is another very important way that a perinatal stroke can present itself. And it might be ischemic, arterial ischemic, as illustrated by this very large area of encephalomalacia involving a middle cerebral artery territory in a child who presented 3 to 4 months of age with hemiplegia, or it can be a subtype that we call periventricular venous infarction. And so there are these many diverse paths to a child having sequelae from stroke in childhood. And so one might wonder, well, how common is this? And overall, it's about 400,000 per year, and this is data mainly from developed countries. We don't really have good data from low-income or low-resource countries. And as I mentioned, about 60% are ischemic and 40% are hemorrhagic. And then there's a very important age-related distribution, and neonates are the most susceptible. And the rate of stroke in neonates is about 10 to 20 per 100,000 per year, which translates into about 1 per 4,000 term newborns. And then it spreads out, and you have a peak in the preschool years, and then a little bit of a lull, and then a smaller peak in teenage years. And it's our thought that these strokes that present at different ages probably have different spectrum of causes. They manifest in similar ways, but in not exactly the same way. And we think that the needs for their rehabilitation and the biology of their recovery is probably a continually changing substrate, which is fascinating for neuroscientists, but also a big challenge. And I think as we design trials, we have to really understand the underlying physiology and get a much better grasp at what the causes are and what therapies and targets that we need to approach based on an understanding of the developmental maturation of these injuries. Overall, if you take the median age of onset, it's about 5 to 6 years, and gender also plays an important role. And also, Louise alluded to this a bit, that not only are boys more predisposed to suffer from ill consequences when they're young, but boys are more commonly afflicted, with about 60% of all subtypes in all age groups affecting boys compared to girls. And then there also are racial and ethnic susceptibilities, and children of African American origin have a greater incidence and greater sequelae. And this is probably for many different causes, and not just from sickle cell anemia, which is usually the first thing people think about as the basis for African American children having a greater prevalence. So what does cause a stroke to occur in a child? The British were really thought leaders in this area, and they collected data from about 212 children from the 1980s and 90s, and looked at them from the point of view of what is that child like when they have their stroke. And they were able to see that about half of children were already known to have some sort of acute or chronic illness, which might have provoked a stroke, but the other half were occurring in children who are seemingly healthy. And this, of course, is always the tremendous shock to parents and providers that their seemingly healthy child all of a sudden out of the blue has a stroke. How can that be? And what are the causes? And so for the last 10 or 15 years, much of our efforts in the field have been devoted to understanding what the causes are as far as we can understand them. And in children who have chronic known or acute illnesses, the biggest underlying feature or cause there would be children with congenital heart disease. And so they make up about a third in this children with what we might call symptomatic stroke. Sickle cell anemia was certainly a big player, and this slice of the pie, now 20 years on, would probably be much smaller because of the success of primary prevention, with early detection of high-risk patients with sickle cell anemia now going on to preventive treatment. And beyond that is a whole kind of potpourri of conditions, things like acute infections such as meningitis, brain tumors. More and more we're finding that certain children with genetic diseases are prone to having vascular complications. But how about this 50 or 55% that were previously healthy? What caused that child to have a stroke? And it wasn't until we had the capability of doing extensive and comprehensive testing with greater technology with imaging, such as with vascular imaging, that we begin to understand more about this. And so in those children who are well investigated, where they're found about 2/3 of them to have something wrong with a blood vessel. And it sounds rather primitive to think of it that way, but that's really about where it started as far as defining these lesions affecting the blood vessels. And so you'll see in the literature terms like arteriopathy or vasculopathy, and we're just, I think, beginning to parse out the different types of vasculopathies that affect children. And in that same data set, it became clear that acute, seemingly minor infections seem to play a role either in the underlying cause or in precipitating a stroke in a child who had some otherwise previously invisible risk factor. So some kind of relationship of infection and vascular changes or vascular abnormalities began to emerge and has been the subject of a great deal of work. And we're learning more and more about these vasculopathies as time goes by, but there's still a great deal to learn. And so it's very interesting to me to hear about all the work being done about in regards to the immune response and the inflammatory response in the brain. And so that ties in, I think, very rationally with this relationship we're finding in children between a recent infection and a vascular lesion. But also, I think we need to look beyond the initial cause and incorporate knowledge that many of you have contributed to about the post-stroke inflammatory response and the cascade of neuronal injury and how we can apply that in children. Certainly the immune system, just like the brain, is a continually changing landscape in childhood. And so it's as though we have to study 3 or 4 different species because the child is a continually changing biology over time. So hang on to your seats and help us. I'll be coming to you for help and hopefully many of my colleagues to travel that journey. Then I'm going to turn now more to outcomes. And certainly it's widely held that children recover better from stroke than adults, and I think there's a lot of truth to that. At the same time, they live with the consequences of their stroke for a while. So when one looks at the overall lifetime impact of their event, then it takes on a different meaning. One might start first by asking, well, what is the survival rate? And the good news is that almost every child survives a stroke, whether it's a hemorrhage or an ischemic event. At the same time, it remains among the top 10 causes of death in children in the US. And there is still some mortality that goes with it, about 5% in ischemic stroke. And it may be as high as 25% in intracranial hemorrhage. That number really is determined by access to healthcare and rapidity of response. Intensive care and neurosurgery and imaging has risen to a level that almost every child can survive if they get adequate care quickly. And then how about the outcomes? And there have been lots of studies that have looked at the spectrum of outcomes, and the overall average of these kinds of studies is that about 60% have some form of permanent disability. Permanent is really lifelong. And many children are still able to function pretty well, but they don't function in the same way they might have if they had never had a stroke. Another important element of this, again, gets back to the maturational nature of this condition. And a child who has a stroke as a neonate doesn't even demonstrate they have antiparesis until they're 3 to 4 months old. So that newborn is growing into their motor deficit. But more importantly, they grow into their cognitive and behavioral deficits and their epilepsy. And we can't really appreciate the full impact of that stroke on their cognition, learning, school performance, and behavioral health until they're in school age. So that may be partly because that's the limitation of our instruments, but it may also be that there is a continuous evolution of the underlying networks and connectivity and the response of the growing brain to injury, where there is in fact loss of developmental capacity over time due to continuous changes of a negative nature. So I think this again is an area that's fertile for a better understanding. And something that's probably more impactful in children than in adults is epilepsy. And overall, in children who have a stroke outside of the newborn period, the prevalence or the later incidence of epilepsy is fairly low, about 5%. But in children who have a stroke around the newborn period, 20 to 40% over their lifetime will have epilepsy. So epilepsy is yet another major morbidity in early childhood stroke. And yet we're not quite done, and children are also prone to recurrence. Children who have an ischemic stroke from many different causes may be at risk for recurrence, up to as high as 15% by 2 years after incident stroke. And children with a hemorrhagic lesion, and most hemorrhages in childhood are not due to hypertension, but they're due to some sort of vascular malformation. And if those vascular malformations are not cured or treated, then they're at risk of bleed. So we have to think about recurrence and preventing recurrence. Now, I already alluded to the fact that it's quite complex and difficult to measure outcomes in childhood stroke, and a number of tools have been developed, which I'll run through a little bit, but still it's pretty difficult to think of how one would measure in a reliable, sensitive, very specific way the outcome of a stroke in a population that includes children who had their first stroke at age 2 compared to age 6 compared to age 15. So it's a really big challenge. And I think we're still learning, refining how we do that. And we can take the long view and measure outcome from these sort of global measures like this instrument we call the pediatric stroke outcome measure, or a pediatric version of the Rankin scale. But that really doesn't tell the whole story. And when one comes to think about, when we come to think about designing clinical trials, we think we have to think very carefully about measuring well what really matters in terms of the long-term outcome, and not necessarily put all our eggs in the basket of one global, relatively gross measure. And I think this is perhaps even more important in childhood stroke than in adult stroke. So we think about the global outcome, are they walking, talking, going to school, and then we think about behavior. We have to think about domain-specific measures, and these also will change with age. Measuring motor outcome is not just some absolute level of function, it is the child's function relative to where they need to be for their age. The same for language, the same for neurocognitive and social and behavioral. So we really have to be artistic and creative and thoughtful when we look at stroke outcome in children. So this just tells you a quick look at this pediatric stroke outcome measure. It was really designed by Gabrielle de Wever, and Gabrielle de Wever is one of the founders of the research network, and she was started her life as an occupational therapist. So she brought that sensibility and that knowledge to her training as a neurologist and her interest in stroke. And so she designed this scale that divides domains into sensory motor, right and left, language, expressive and receptive, and a sort of global cognitive behavioral deficit. And each domain is scored by an examining neurologist on the basis of a general, typical, detailed neurologic exam, and gives a score in each domain of 0 for no deficit to 1 or 2 for mild or moderate or severe deficits. So this has become now kind of the industry standard for a global functional and neurological assessment for children who've had a stroke. It's done by a neurologist and scored by a neurologist based on their own internal knowledge of the developmental norms. So that's intrinsic to this measurement. It relies on a neurologist who knows children. And the Rankin scale is similarly adapted. Many of you who are familiar with this will recognize that this is really kind of a global ranking of 0 to 5 for mild, moderate, or severe deficits. It's widely used in Europe and is often used in by a variety of providers, and it can be, again, give you a fairly gross, but at least one way of grading outcomes. So what do we know? What are the outcomes of stroke and in children? And the reason I'm showing this is not just not really to represent the full data that we have from the literature, but really to illustrate the variability. And this is the first line is a cohort from Europe of only 37 children. Median age of onset of stroke is about age 4, and all of these studies seem to pick up a similar cohort of children whose average age at onset of stroke, and most of these are non-neonates around the early school age years. And as you see here, the mortality is pretty low for most of these children. And people use different ways of measuring outcomes, and they found, for example, neuromotor disability across a wide range of frequency. In one case, only about half, in another case, 2/3, in another case, almost 80%. So is this a true representation of variability, or is it just variability in our ability to measure? Cognitive and behavioral outcomes also vary widely. In this top level, they found a generally lower IQ, and none of them had aphasia. On the other hand, the subsequent group, this large group from Canada, almost a quarter of them had some sort of language impairment at their follow-up. And likewise, the United Kingdom, British cohort, about 40%. So it seems as though we we need better measures, we need larger groups, we need more representative groups. I think the take-home message is that children do have long-lasting and diverse disabilities that range from neuromotor to language to behavioral deficits. One of the most recent, probably best characterized group is from the Canadian cohort, and this was just reported out a couple of years ago where she had almost 1,000 children. And this represents both perinatal and childhood stroke. About a quarter of this group were children with perinatal stroke, and as is typical, a slight preponderance of boys, and their median interval to follow was 3 years. And these were children who almost always survived their stroke, about 5% mortality. And one might wonder, where are the strokes and what circulations are involved? And this is pretty typical of childhood stroke cohorts. The majority, in this case 67%, involved anterior circulation stroke, but posterior circulation stroke may be a somewhat more overrepresented location for stroke in children as compared to adults. So when we look at outcomes, we have to bear in mind that the spectrum of injury and outcomes would certainly be different depending on what circulations and where in the brain the injury is. And when we look at kind of pool of data, it really hides a lot of detail and a lot of variability within a cohort. And this shows you what the different subtypes or causes of stroke. Similar to the original UK cohort, almost half of these children had something wrong with the blood vessel, or what we call an arteriopathy. About a quarter were thought to be cardioembolic, and then about a quarter are what we call cryptogenic. And this is a subgroup that we define as having no apparent cause, even though full investigation has occurred of the heart, blood vessels, and the thrombotic systems. And what this shows below is the kind of broad sweep of outcomes where the stroke outcome measure was normal, shown in green, mild or moderate in light green or light blue, or severe in dark blue. But what you see is if you count the children who were just completely normal, the numbers look pretty similar to what you saw before, but the message is to either be the glass is half full or the glass is half empty. When I talk to parents, I like to present the glass as more than half full, and that is that your child will make very substantial recovery. And that I am sure of in almost every case, that even if that child comes in with what looks like to be a very severe stroke, they are going to make a lot of improvement. Now later down the line, when we sit and talk and the reality hits, then we are talking about there are going to be long-lasting, subtle, mild, or in the small number of cases, perhaps 9–10% of very, very severe deficits. And then you have this middle group that 11 to 20% will have epilepsy. So this kind of is, I think, a nice thumbnail sketch of what the outcome looks like and the challenges that we have for helping these families and understanding better. Do we know, for example, whether the stroke subtype changes your outcome picture? Well, it certainly changes your outcome in terms of recurrence. And does it matter if you have, say, congenital heart disease, and all of the morbidity that goes with congenital heart disease may impact your recovery from that stroke? So these are factors and nuances that come into play in terms of understanding the neurobiology and helping to manage them as they go through life.
So Becky, in those kids who have epilepsy, is that thought to be a selective loss of inhibitory interneurons in some of those kids, or why do they develop epilepsy?
Well, it's a very good question, and there must be something age-related because the prevalence of epilepsy is much higher after perinatal stroke than after childhood stroke. The biology that's been very poorly worked out, we know that the bigger the infarct, the higher the probability is that you'll have later epilepsy. So I don't know that we can really say it's a difference between loss of inhibitory systems versus excitatory systems. I think it's just a loss of the normal development of the balance of networks and network connections. There also are thoughts about whether children who develop epilepsy might have some kind of genetic predisposition to have epilepsy. So are there epilepsy genotypes that might make a child with a stroke more likely to have epilepsy. These are all unexplored frontiers. So bring on the science. Another way of looking at the outcomes is in terms of IQ and intelligence. And again, here is a story that I like to present to parents as the glass being way more than half full. And even if a child's had a very large infarct, they are almost always going to have intelligence broadly within the normal range. So their child's not going to have major intellectual disability, but they are going to have challenges with cognition, learning, or behavior. So the mean IQs have been studied in some of these cohorts, and you'll see in the perinatal stroke group, for example, in this cohort, the mean IQ is 91. Certainly that's within the normal range, but that child's brain has taken a hit, and they're paying a price in terms of cognitive function. As compared to, say, the child with early childhood stroke, they're still very well within the normal range, and they've taken a hit, and it's less of a hit as compared to a teenager who really has been able to preserve their intelligence better. And it kind of makes biological sense, but one would sure like to better understand all that biology better so that we can think about interventions. Our group looked at more specific neurocognitive profiles. We brought back a group of 40 children who were between age 3 and 16 years who had a history of perinatal stroke to get a better sense of what is the overall cognitive profile, and are there certain types of cognitive functions that are more susceptible to injury than other cognitive functions. And this shows you that the batteries they did looked at full-scale IQ, measures of attention, working memory, verbal recall, inhibitory abilities, flexibility, organization, and the word yeses. But you'll see there's a variety of changes, and the areas of function that took the biggest hit were what we might call executive function: attention, organization, ability to inhibit. And so kids who have recovered from a stroke as a newborn frequently will end up having a label of attention deficit disorder. And that's not your garden variety attention deficit disorder. It is something that was related to their underlying injury. And so these are long-lasting, lifelong, really life-changing problems that these families and children have to deal with. And again, if only we understood that biology better, perhaps we could mitigate the impact of that over time as the brain develops. We certainly have to manage them when they come to us.
Yeah, you got about 5 minutes.
Okay, so I better hurry up. So do children really recover better than adults? Maybe, not so much. So one has to really control for the factors that predict outcome after stroke. And this was a nice study from the Swiss group looking at 128 children and compared with 200 adults. And they controlled for the stroke severity, and they looked at different stroke subtypes. And what they looked at was the favorable outcome of children at 6 months compared to adults based on the MRS being 0 to 1. And in fact, children were about the same as adults. If you controlled for anti-stroke scale and you looked at the percentage of patients who had an MRS of 0 to 1, it was pretty similar to young adults. So certainly that's probably still better than a 60 or 70-year-old, but being young doesn't necessarily guarantee that you're going to be better than if you had it somewhere later. So what are the predictors of outcome? I already alluded to the fact that age at onset seems to be important. And it may be that if you have a big stroke as a preschooler, that might be the worst time of your life to have it. The infarct size, the severity of the initial clinical stroke, all of these are predictors for outcome. And this is a slide that I love to show, and that is that neurons live and die by their connectivity. The people in this room know that very well, and it's especially true in the young brain. This shows an MRI with diffusion imaging. In day 3 compared to day 10, and this is an example of a cohort that they looked at. And what you'll see in the parental tract, the descending corticospinal tract, at day 3, you don't see much injury, but by a week later, there is very extensive downstream degeneration in that corticospinal tract. And this is very common. We see it in almost 100% of the newborn strokes that we see. So there's this connectivity-driven dying back of connections that is very much age dependent. So this really makes us think about the substrate for intervention. We have to think about where is the infarct, how big is it? Is it just involving white matter? Is it involving the entire cortical spinal tract from the cortex down through the internal capsule? Does it affect sensory and motor systems? And how can we think about our interventions relative to that? So trial design, I think, and plasticity are driven by activity. And again, I don't have to tell the people in this room about that. But I think it's probably even more true and more important in the child with recovering from a stroke, that there are probably windows of opportunity that really could maximize recovery if we can intervene. At the same time, there's probably a much longer period of time during that child's life when they still have the capacity to rewire and to reconnect. And also become sensitive to the fact that we need to think about both motor and sensory systems. And again, I think people in rehabilitation are aware of that. And I think stroke neurologists are probably not as attuned to that because we don't spend as much time thinking about the sensory side of things. But if you don't have any sensory inputs, then you can't drive the how that guides the motor function. And this is a nice study from a colleague of mine, Alyssa Newport, who is really a linguist, she does the neuroscience of language, and it shows how language recovery after stroke is extremely age dependent. And she studied children who had stroke in very early infancy to test the theory that both hemispheres are equally capable of supporting language development. And if you then look at language development in a group of children who had a perinatal stroke, you might theorize that if their left hemisphere was affected, they might be worse than if their right hemisphere was affected. And she found that was not the case. It didn't matter which hemisphere you had your stroke as a newborn, you were equally likely to restore that. And this shows you how that happens. And she did functional MRI in children who had a perinatal stroke, and what you can see in the controls is the typical localization of language function, and what happens in the survivor of a perinatal stroke is the homotopic regions are taking over that function and are able to largely recover. That isn't so true at the 2-year-old or 5-year-old or 7-year level. So what do we do next? Motor learning therapy, I think, is really the next frontier. It's been done by your colleagues and friends here at Blackdale and Andrew Gordon in Columbia, and they're really, really important leaders in that. And I think finally this is coming to us in a bigger way in childhood. There's constraint-induced movement therapy involving restraint of the good limb. But then more recently is this bimanual approach, because children really use both hands in doing almost all activities. So both approaches seem to be similarly beneficial. And this is just one example of Andrew Gordon's, one of his trials, looking at this type of intervention for children with hemiplegic CP, which includes many children with perinatal stroke. They have to receive treatment. They receive treatment for 6 hours a day, 5 days a week for 2 weeks. That's a big deal for a child and for a family to undertake that kind of treatment, but it works. And they found sustained benefit after for a month later. And many of these kinds of interventions are still very early in the very early stages of development. This compares the constraint-induced versus the bimanual, and you can see from pre to immediate post to 6-month post that both approaches produced important gains. And I'm going to skip over these because these are kind of intuitive. There certainly is interest in the pediatric realm to incorporate electrophysiologic interventions as well, and Adam Curtin is one of the few in our world who's doing this, and he's doing it with quite a bit of success. He did it initially with TMS, and now he's doing direct cortical. And he has also shown that in children who have an electrocortical intervention along with the intensive motor learning, they can make important improvements. and he's doing that with direct cortical stimulation as well. But these interventions are just beginning to gain traction, and one of the really exciting things that is around the corner now is an NIH-funded trial, which I'm just going to jump to. Oh, I might say this could also be useful in aphasia, and Adam Curtin's group in Canada has also tried intensive therapy with TMS for children with aphasia following perinatal surgery, and it's also seen improvements. So I think we're on the threshold of hopefully a revolution in how we can do this. And one of the things that we're excited about is this I Acquire, which is a intervention at a very young age, children 8 months to 2 years. That's at a time when motor systems might still be even more plastic than at a later age. And they will receive constraint-induced therapy 3 hours or 6 hours a day for a month. I just went to an investigator meeting, and one of the lead investigators says, think of this as training for the Olympics. It's not just a therapy you do once or twice a week. It is really intensive training, and I think that that's the way we have to think about this. So questions and observations, grist for the mill. How do we individualize based on anatomy, based on maturation of the child? How do we think about the most important outcomes, quality of life? We have to really think about language and cognition. And my experience with kids has been is that they can adapt pretty well to a motor deficit, but the really life-changing impairments are language, cognition, and behavioral problems. And those are areas that we haven't even begun to scratch the surface. How do we get a durable effect? Do we do this individually? Do we incorporate therapies into groups? Do we do it in the home, institution? I think the answer is we need to do all of these and all of the time. I'm going to skip over this and just take a kind of a parting word from Adam Curtin, where he, I think, learned a lot from his kids. Kids, I think, are more social, herding individuals than adults, and they need to function within a group and probably get benefit from being in a group. And so what he says is that the psychosocial benefits of being in this kind of intensive therapy derived not only from the individual effort that they put into it, but from being part of a group and feeling like you belong, and how each child kind of measured himself or herself against the group, and they egged each other on, and they competed with each other. So that's another thought that we have to keep in mind. I think it's unique to children, and it's potentially something we can leverage into a good benefit and interest.
Great, thank you very much. One quick question. Kathleen.
Hi, so I'm asking this question on behalf of Dr. Friel. Can you address the interaction between motor speech and cognitive outcomes? Can cognitive testing be biased by a child's challenges in motor and speech, and how would you control for that?
That's a very good question, and I think neuropsychologists who work with children who have a variety of deficits take that into account, and they will rely on the measures of cognition. For example, in a child with aphasia, they will rely on nonverbal output measures. At the same time, one's estimate of intelligence is still reliant on each stream of function. So whatever number or outcome measure we get from such a modified approach needs to be interpreted with caution. But really good, well-trained neuropsychologists and child and psychologists understand this, and they take it into account in their testing and in their reporting and interpretation.
Great, thanks very much. Our next speaker is Melina Dawson, who's the Director of Neurodegeneration and Stem Cells at the Institute for Cell Engineering at Johns Hopkins. As many of you know, Melina has been a member of our scientific advisory board here at Burke for the past several years, and in that capacity, she's provided needed scientific and strategic oversight one of the reasons why I asked her to join that group was not only does she do beautiful work that's impacting not only stroke, but Parkinson's disease and ALS, but I've always been amazed at her the fact that even though she didn't go to medical school and get training, that she has a remarkable understanding of the clinical problem. And I remember when I was in Baltimore, often hearing Valina on the radio talk shows talking about stroke. And I thought, wow, how does she know all that clinical detail? And I attributed it more to her brilliant mind than the fact that she was married to a neurologist. So,
thank you very much. I do get coached very well though before I go out. But certainly the clinical problem has never been far from our mind. Doing basic science research is very exhilarating. It's always fun to see something new. But my grandfather was an oncologist. He trained at Hopkins and then at Stanford, and he discovered the treatment of thyroid cancer that's still used today. And he told me as I was growing up that I could be anything I wanted to be because I was here in the US. And that he encouraged us to do whatever we wanted to do. But we had an obligation because we had pulled the brass ring just by being born into this country, that we could do what we wanted to do, so we had to give back. And it could be something as simple as volunteering, but whatever we did, we needed to give back. And so because we really enjoyed research, we devoted research to understanding neurologic diseases with the hope that one day we can give back. So I am a PhD, I'm not an MD, and I've been studying mechanisms of strokes since the 90s, very early 90s. That's actually ended up meeting Ron. And most of the work we have done and have done has been on acute stroke, but recently we've also been considering chronic stroke. Because as you've been hearing from all of the speakers this morning, that you have this window of opportunity very quickly. You know, Bruce was saying, get into the hospital fast. But what if you can't get into the hospital fast? What do you do then? And can you, even if you can get reperfusion, there still is some reperfusion injury. So even though there's a population of patients that do really, really well with reperfusion, there's another population of patients that don't experience that same benefit. And so we've been thinking about, could you do some neuroprotection during those critical window periods? And then I'll tell you a little bit about what we're starting to think about, how you might address chronic stroke. So these are the concerns that we have. So the unmet need is the neuroprotective therapies that could be possible. For a number of years, over a decade, the NIH decided to rather ignore neuroprotection and focus on other issues. Neuroprotection is really done in the setting of Parkinson's disease. We need to have neuroprotective therapies during the reperfusion phase, as well as hopefully some neuroregenerative therapies to get the brain to start to regrow. And then if you can't do those two, or you've maxed out those two approaches, possibility of neural replacement or neural restorative. And this is something I thought would be hokum, but at the end of the talk, I'll show you there's been some surprises. I was certainly surprised. So over the last almost 30 years, investigators across the world have really defined molecularly the elements that occur immediately following a stroke, within minutes of having a stroke, following hours and days. So depending on what your target is, if you have a target that's going to be available and important within hours of a stroke, that's when you need to apply the drug. You shouldn't apply it hours or days or weeks later, which had been what was being done, I think, in the early 90s in stroke trials. And then the trials, of course, didn't work, and then people lose their interest in trying to develop therapeutics and to develop drugs for an injury. So when I was a postdoc at Hopkins, actually I was a postdoc at the NIH, but Saul never allowed me to study. He's not here, but I was a postdoc with Saul. We discovered that glutamate excitability. So glutamate is used to, when you move, when you're learning,
But when you have too much glutamate released under certain settings, the brain dies. And we found that the reason that this happens is that the glutamate receptor activates this enzyme, neural nitric oxide synthase, which exists in almost less than 1% of your neurons in your brain. But it's in the critically important neurons, and it generates this molecule NO, which then diffuses around other cells, which are also now very active because they're getting a lot of glutamate. And you end up having an oxidative stress experience in which peroxynitrite is generated. And this peroxynitrite does two things. One that we published well here for many years, it damages DNA and activates this enzyme PARP, but it also activates this molecule C-able kinase, which is important in certain cancers, and there's drugs for this now, some that get across the blood-brain barrier. And C-able phosphorylates a member of the protein translation machinery, and it gets a gain of function, and it moves into the nucleus where it directly activates PARP. So we have two ways to activate this protein PARP. And when PARP gets activated, it generates this molecule poly-ADP ribose. And poly-ADP ribose has many functions within the nucleus. It regulates what genes get turned on, what genes get turned off. It regulates DNA repair. And it can get carried out of the cell by what we have here is an acceptor protein. And in a paper that has been ready to submit for publication for the last 6 years, this person left and went and started a company, and we told her we're not signing anything anymore until she actually submits the paper. We know that this acceptor protein is a histone protein. And so it gets out of the cell, and it goes back, there's mitochondria around the nuclei. The mitochondria have a hexokinase, which is affiliated with the membrane. When it binds PAR, it moves away from that membrane, so then the mitochondria depolarize a little bit. And then PAR also binds to this unfortunately named protein AIF, which stands for apoptosis-inducing factor. It never induces apoptosis. That was an artifact of its discovery. But unfortunately, we were this name. So when we name proteins, we use gods, and we don't use function, because they always have something else that they do. So when AIF has PAR bound to it, it picks up another protein in the cytosol that has a day job as a cytokine. This is macrophage migration inhibitory factor. But when it picks this up with the PAR, the two move to the nucleus, and there the two bind to DNA, and they chew the DNA up into great big pieces. And that's pretty much the end stage of cell death. So at this point, the cell's now committed to death. And then there's some things that can block cell death, like Meduna and PAR, but we won't talk about those. So we spent a number of years building the signaling cascade in stroke, and then found that it actually was important in Parkinson's disease as well. So we wanted to know a number of agents and targets had been developed in mice and rodent systems and culture systems, but they, when they went to move into the clinic, they weren't very successful. And so there were several possibilities. And one was that all of my colleagues sitting here did not run good clinical trials, and so we figured that probably wasn't the case. There was this concern that maybe mice and men are different, and this is certainly the case. There are certain neurons that we have in our brain that mice do not have. And so we wanted to find a system where we could really study these mechanisms in real human neurons, so we could develop drugs and treatments in real human cells, because oncologists have just been blazing away. But when you have a tumor, you don't want it in yourself. You're very happy to give it to your clinician and let them study it. When you're losing cells in the brain, you want to keep what you've got. So we started thinking about what were the differences between mice and men. There's a lot. The structure of their cortex is a little bit different, but not so much. And so we said, oops, wrong one. Let's see, now this is going to be motor coordination. Who's doing motor? How do I get out of this? There you go. Oh, I'm still, yeah, I'm a PC person. And so we wanted to develop human cortical cultures. And at the time, and still actually, the problem is you can generate, differentiate excitatory cells or inhibitory cells, but most people can't do both. And so what they end up doing is taking cultures of each and trying to mix them together. But that doesn't work, because we know that you wire together as you develop, and things work nicely. So if we look in the literature, still, most of the cells that are out there don't have this complex network. And the brain organoids, as far as I know, none of them have this complex network. And they also don't have microglia, because microglia differentiate from a different set of tissues altogether. And microglia are very important. So, shoot, I did that again. Let's see. I just forwarded. No. Okay, so the reason you need a balanced network is so the cells will talk to each other both physiologically, and then when you introduce the pathophysiologic signal, that they will respond in a way that a normal cortex will. But more importantly than anything else, these cells, these cultures have to express that 1% of the neurons that express nNOS, because if they don't, you can't kill the cells. You can't do a stroke in a dish, you can't do schizophrenia, you can't do Alzheimer's. And so this was important to us. So, you know, so what one of the fellows in our lab did, is he went back and he looked at the development of the cortex from in mice, in neonatal ranges, and what happens at each stage. And he realized that there were two sets of events that occurred that were important to get the entire cortex. And taking advantage of that, he developed this system or differentiation protocol that we call RONA. So we take human and embryonic stem cells and we start developing them in culture, and they form at some point these what they call rosettes. And these are just clusters of neurons, neural progenitors, glial progenitors, and then these little light blue cells out here will never be anything. And in the fluorescent images, you can see them very nicely, that even under a light microscope, you can see the difference between these cells. So he just takes a scalpel and he excises these cells. So he's sure that he doesn't have any of these other light blue cells in here. And then gives them a time sequence of different growth factors and such. And so what we end up with is a culture that expresses both inhibitory and excitatory neurons in the proper percentage. So we have both GABA and glutamate containing neurons. Gosh, I should have trained for this before. But more importantly, the inhibitory neurons, which have a number of different types of classes of neurons, are all expressed in these cultures, and they're expressed at the same percentages as if you went into a human postmortem brain and you counted the number of inhibitory neurons in a block. So we now have a nice balance of excitatory and inhibitory neurons. And so this is the one I want to point with. And then all of the layers of the cortex are represented. And here we're just doing immunohistochemistry for markers of different parts of the layer of the cortex, based off, but we've also done a lot of RT-PCR and other kinds of validations. When you, got it right this time, when we give them a little bit of structure, these cells try to form a brain. And so they start developing into various layers of the cortex, which is kind of cool. And then you can think of, my first thing is I thought of all the different experiments we could now do looking at physiology and such, in addition with these cultures. So the guys in the lab really wanted to do the organoid kinds of studies and start looking at the three-dimensional interactions and start putting microglia and such in there. And I told them that the organoids were just a mess. You can't quantify anything in the published literature. There are these blobs, there are different sizes, they've got little poochy things that pop out. And so I said, you've got to figure out a way that we can have a uniform structure with uniform layering that we can then go in and quantify, almost do like stereology in an organoid. And so what they developed were these little hanging clusters. They're hanging drops, which are kind of cool. And they actually don't fall down when you do the stuff you have to do culture on. And they form, these organoids form into the layers of the brain. So we now have like these little mini brains in a drop. And so I don't have the movies because it was hugely large, but we are now culturing microglia and putting them on the edge of the drops and watching them move into the interior of the organoid. And then we can do things to cause disease in the organoid, Parkinson's disease, and make it ischemic, and see what happens to the microglia. And we can also engineer the microglia to do or not do what they're supposed to do. So with these cultures, the other thing that they need to do is they need to behave physiologically like a brain. And so what happens in brain cells is you've got neurotransmitters coming down through an axon or through a dendrite. They get released and they stimulate the cell that's next to it. So you can record from different parts, the cell body or down in the terminal region, and you should get these certain kinds of recordings. And these will develop over time. So an immature brain behaves very differently than a mature brain. So we followed these cultures over time. And they do everything. By 12 weeks in culture, they have fully mature ion channels, voltage-gated channels, their minis, their inhibitory and excitatory signaling is mature. But most importantly, and this is still the only paper I've seen, they're the only cultures that homeostatically scale. And this is the process that's involved in learning and memory in patients, or in people. And then because they also have Nosson neurons, we were hopeful they would die. So we're at this point and we go, okay, now can we do stroke in a dish, or have we just wasted a ton of money and time generating cultures can be totally useless to us? Fortunately, they do die if you put glutamate onto them. So glutamate is the molecule that initiates the damage during a stroke. And we see very nicely that we get a dose-dependent increase in glutamate in these cultures, as well as a time-dependent increase in toxicity. The only difference between mouse and man is it takes a little bit more glutamate and it takes a little more time. So instead of a 5-minute application, I think here we were doing 15 to 30 minutes application of glutamate. So you need a little higher insult, but then everything else is the same. If you block the NMDA receptors, you get a complete protection. If you block the AMPA receptors, you get a little bit of protection. This looks just like 30 years of work in mice. With the NMDA, again, we get a nice dose-dependent increase. This is because most of the injury is mediated through this receptor. And then we looked at stroke in the dish. And so a 60-minute OGD in a rodent culture would give you about 60 to 70% cell death. We get that same with about 90 to 120 minutes of injury. If we put on inhibitors of apoptosis, necrosis, and autophagy, we really don't see any protection in these cultures, and we've never seen any protection in our rodent cultures, so that was consistent. But if we put in inhibitors of nitric oxide synthase, an NO inhibitor or a PARP inhibitor, we see almost a complete protection. So we went home and slept soundly that night, knowing that we didn't have to recount all of our work over the years. We see a very nice induction of PARP. So PARP, when it gets active, it generates PAR, and we can measure that using an antibody. And so with NMDA and or OGD, we see an expression of PAR, which is blocked with both NOS inhibitors and PARP inhibitors. And these are all experimental compounds. If we knock out PARP in mice, we have almost no infarct volume in mice. So we said, what if we knock out PARP in our cultures, these human cultures? We're using CRISPR-Cas9 here. Can we protect them? And so again, just like with the PARP inhibitors, we get a complete protection by knocking out PARP activity. And that's just an experimental tool. Now, PARP inhibitors are being used in oncologic purposes for breast and ovarian cancers, and for reasons that the companies will not tell us, they know that some of them actually do cross into the blood-brain barrier. We confirmed that in mice, and so we took a battery of these clinically used PARP inhibitors and put them on our culture dishes and showed that they block stroke in a dish in both our cultures for male humans as well as our female cultures. So we get a nice protection. So hopefully this is going to be useful. Now the next step in the process that I told you about was that we activate this cytokine MIF to move into the nucleus and become a nuclease. MIF knockout mice are resistant to stroke, and so we thought, well, since this is a gain of function, this might be a really good target for developing a molecule to treat stroke. And so we did a screen with the oncology department, and not because just happens they had an interesting library. And we found a compound that blocks DNA cleavage and is neuroprotective, but does not block the the inflammatory components, the part of the cytokine activities of NIF. It took us a few tries, but we found a compound that gets into the brain very well. And when we give it, in this case, these aren't strokes because we were going quickly for commercial concerns. But this is a NIF knockout mouse that's been injected with MDA. So it could have been glutamate, that glutamate gets taken up by glia, so we used MDA instead. So the wild type, you see a lesion. The white is all the damaged tissue from the injection. The NIF knockouts, you see no lesion. If you look at our compound at both at 15 mg per kg, this is the one that gets into the brain very well. You see just a tiniest bit of lesion at 5 mg, and you see absolutely no lesion at 15 mg. This would be an aspirin-sized pill if it was given to a human patient. So we're currently, we've identified and defined the binding pocket for NIF so that we can do computational biology for our small molecules, as well as continue to develop this compound. So we think that parthenoidos, we still are big believers in parthenoidos, that it's an important pathway in neurodegeneration and in stroke, and that if you can block this, this process, that you can elicit neuroprotection. So the other thing we want to look at quickly is preconditioning. If you take a tissue to the brink of death, but you don't let it die, you come back 24 hours later and these tissues are remarkably protected. And so the question was why? And we had done some work in mice, but nicely at the time that we got our human cortical cultures developing, there was this ability to do high throughput, deep sequencing, RNA sequencing. And so I have a fellow in the lab who works very hard. And first he showed that we did get preconditioning in these cultures. So these are cultures here that are naive, they die. If you give them 30 minutes of preconditioning and then come back with the OGD, they're protected. So he did deep sequencing and RNA sequencing on these cultures, starting at 15 minutes and going for 72 hours, like every couple of hours. And we finally got all the data. You can see there's a number of genes that go up, a number of genes that go down. And we get all these variety of clusters. So in this group, there are early immediate response genes. Did I get that right? Because you've worked with Javier.
Yeah.
That were previously not known. They're human specific. Yes, immediate early genes. Thank you. They're previously not known. But there's a number of different clusters. We've got probably 100. We just finally got them all uploaded onto the databases, so they'll be searchable, and we will get the paper submitted soon. From this cluster analysis, we've identified a master regulator as one protein, and it regulates CREB, NSF, all of the neuroprotective transcription factors. And so we're thinking that would be a good gene to go after to elicit protection in the brain. So the last is, I just want to briefly mention cell replacement, because there's a lot of charlatans doing cell replacement. They just have a cell and they plop it into the brain and they think it's going to work. The ones that are currently being tried are mesenchymal cells, that's spinal cord precursors, and cancer cells. I don't know why. What we need to do is have the right cell for the job, have blood supply myelination, integration, and long-term survival. So we've been fiddling around with this. We inject neural precursors into the brain. Not only do they survive, which was a surprise, they integrate. We get very nice physiology from these cells. If we have a small stroke, they will go into, expand into this stroke volume and make contacts. And this was a very fast study. It's not the way we're doing it now, but these animals that got these cell replacement therapy did better behaviorally. This is a motor task of reaching and grasping. What we plan to do, what we are doing, is we're going to combine microglia into those organoids before we transplant them. And then we're not going to transplant immediately after the stroke because that's not useful. We're going to wait at least 3 to 6 months, get full deficits, put the cells in and see if they survive out. So that's in general what I think needs to be done. That's not what's being done right now in the industry, but I think I was surprised by the outcome of these things, so I think it might be possible that there will be treatments for chronic stroke that could be cell replacement therapies. Okay, and of course, I don't do anything, I just write and come and give talks. But we have a tremendous group of postdocs and young scientists, some who started their own labs. People who work with at Hopkins and outside of Hopkins are terrific. I have a lot of disclosures, but basically, you can take it. I didn't talk about any of our drugs that are under development. The only thing, I do have stem cells. I consult for a stem cell company. We're trying to do it the right way, but we'll see. But thank you very much.
Questions? Yeah, here.
There's a reasonable concern of people who've been around for a while, but that is, you do all agriculture, you're dealing with high oxygen. Now that you have all these really beautiful studies of organoids, you're still growing them at a really high oxygen compared to what your brain is. How do you put that in your thinking? Does it just not matter? It seems like a lot of signaling is going to be altered as the cultures are grown at physiological oxygen.
Right. So that's been a long time question, and it's a good question. I think that the high oxygen levels actually help because they age the cultures fast. They make them older faster. And I was concerned about this in the early days when we were setting up the culture systems and thinking whether we had to adjust the oxygen tension within the culture incubators. But we've been able to mimic things in the adult mouse brain, that they've been parallel. And so my concern about that has been lessened just because the same treatments, the same stimuli ended up giving basically the same percentage results. So it may be that we've just gotten lucky for 30 years, but if we'd gotten vastly different results, then I would have gone back and readjusted the culture system.
So Elena, do you think that is also what causes the shift in the dose response? Because you're growing these cultures for a long time, they're essentially being stressed. So they may have created adaptive mechanisms that would resist lower concentrations of glutamate or shorter periods of glutamate.
That's entirely possible because it is a longer period, but it could also be that it's a species difference.
But it's a transporter or something.
Yeah.
There's another question.
I'm just curious, you talked a lot about NNOS and peroxynitrite and superoxide. I'm just wondering if you could comment, if you're not comfortable, that's okay, on the ongoing phase 3s, the 2 phase 3s for the PSC95 NNOS inhibitors, and what you think of that body of literature and how you feel about the likelihood of success for those programs.
So Mike Timiansky's a good friend. I think he's terrific. And I think what he did was just brilliant because he went in with this. So this is an agent that goes in and moves NOS, nudges it off the receptor so it can't really be activated because it's just out of the zone of the calcium influx for full activation. He went in and looked at patients that were undergoing neurosurgery, reproduced this in monkeys, showed that neuroprotection was possible at a time when the NIH and a lot of the clinicians here thought neuroprotection was just a dream that would never occur. And so I think he did everybody a great big service by bringing neuroprotection back as being a possibility. I haven't seen the outcome of those trials yet. I think phase 3 hasn't been decoded, but phase 2 looked like it was doing pretty well.
It'll be reported at ISC 2020.
Okay. I would suspect they're going to see, hopefully they're going to see something that's good.
I'm curious about the comment you made about only 1% of the neurons expressing the NNOS, and whether you think that translates to a clinically meaningful benefit in a non-excitotoxic setting, like in a situation where you have a massive infarct, and there's certainly going to be more than just that 1% that are affected.
So, yes, I understand. So that 1% produces the NO that affects everybody else, right? So without that 1%, you get absolutely no infarct, no matter how hard you have to really hit the mouse hard to get it. So yes, it will affect that because just shutting down that amount of NO, then it gives that cell the time to repair itself. It knows it's injured, but it hasn't hit that kill switch to go in and activate PAR to a level where the cell now just says, I'm giving up, and the DNA hasn't been cleaved. The thing I also mentioned with the myth knockouts, even though all of that bad stuff has happened, you've got stress, you've got the glutamate, you've got the PAR formation, you see PAR go off the root. Because the DNA has not been fragmented irreparably, somehow those neurons repair themselves. The infarcts are very small, and we see almost no DNA damage in those cells, and it repairs if you follow it through. So I'm hopeful for Mike, because I'm hopeful we're going to take these other drugs into clinic as well.
Yeah. So, Mike had data in humans that he could actually reduce infarct size in patients
Yeah,
who are
the
cold.
ECIC bypass trial.
Yeah.
That trial. Yeah. I mean, yeah.
So there is some, I guess, whether that's going to translate into giving it in the ambulance is the question.
Right, but that was a brilliant way to do it because it was quantifiable.
That's right. Great. Any other questions? Thank you very much. All right, so now you get to hear from the home team. This is Dr. Sydney Cho, who's the head of our preclinical stroke modeling here at Burke, and who does also very elegant work on the immune system and stroke. And Sung-hee moved here soon after I got here and really brought to the table a really impressive, elegant, and rigorous approach to modeling physiologic stroke in animals. I think a lot of the stroke modeling that has been done has been done with photothrombosis of a particular area. What's been great about what Sung-hee's been doing is she's been using MCO occlusion, which mimics what happens in many humans, and then following that out to 6 months, really the chronic period in mice. And this has provided incredible new insight into what's actually happening throughout the spectrum of epochs after stroke and how we can think about intervening. So it's great to have you speak.
Thank you so much. This is tremendous pleasure and the honor, as well as privileges to include it as a speaker list, along with all those prominent female stroke scientists. And this is the day I feel really lucky and proud to be a female scientist. So we have a lot of the young faculty members who's been using Keynote instead of PowerPoint, and I always wanted to use it. And then this is the first time I'm using it. It's going to be my test platform. And also because of the something that I cannot use the pointer, so I'm going to also adopt it using my iPhone to navigate it. So if I'm successful, you can tell that it's even my generations who use some of the other technology. So today I'd like to talk about the immune systems, and especially in the all the way through the different stages of stroke, and then how they might be impacting the stroke recovery. And then this is really basic science using the mice. So I guess I don't have to tell much about it. What is the huge challenge that we are having in terms of the translations, because there's over 1,000 targets that has been really validated, but we were really unable to translate it in the human clinical settings. So it means there's a huge gap in the translation. So people has been actually addressed these issues, and some of this is actually outcome analysis. So in animals, and what we haven't really focused is the injury size, and then we are looking at really a period of time. But in human, what is really matters is the functional recovery. So in the to circumvent that issues, is, we are actually starting looking at the behavior outcome in the animal model. Then we should try to narrow down the gap between the basic science and the clinical settings. And the second one is the animal model. I mean, this one has been also touched by the morning speakers. We are using the really male young animals, but again, this is a human. There is many risk factors that could be modifiable or not modifiable. It means the male, female, or young and old, as well as a lot of the metabolic issues, like high cholesterol or the diabetes, obesity. So there is many studies, including us, that we actually started putting those comorbidity in the animal model of stroke. And by understanding them, then we might be able to have a better understanding in the human stroke physiology. But really, this talk is a superficial influence. Stroke has been really considered as a brain matter, brain injuries, but nevertheless, there is a huge infiltration of the immune cells into the brain. So among all those, I would like to really discuss and focus the rest of my talk in the peripheral influence. So one of the really important the question that we have, is there any way that we can actually modulate the immune system? And if we do that, then can we influence the stroke pathophysiology and the functional recovery? And then to answer the questions, first of all, we really need how immune cell changes in the comprehensive context of the stroke stages. When I mean the comprehensive, right after stroke, and you have this acute and the subacute and the recovery. Of course, this is not really defined in time, but this is really continuum. So in the mouse, usually we consider the acute is from somewhere between hours and days, and subacute could be falling into days and weeks, where the most of the spontaneous recovery occurs. And the recovery could be weeks to months. Remember, the rodent, their metabolic rate about 7 to 8 times faster. So in human terms, and if you want to extrapolate it, and this could be actually, so acute could be days to weeks, and subacute somewhere between weeks to month, and the recovery could be month to years. So using this animal and that in these different stages of the strokes, we use the middle cerebral artery occlusion. And this is most widely used animal model of stroke. And then basically, as you see, this is MCA, and then it works close by the critical filament. And what it does is actually mimic the focal stroke in the humans. So using these models, and what we did is we have now almost 10 different post-stroke time points, somewhere from 1 hour to all the way up to 6 months, and including one of the before strokes. With this 11 time point, we actually ran into the bulk RNA sequencing. So this is done by the collaboration with the Regeneron pharmaceutical companies. And this is the result. So as you see here, this is the contralateral side. It means it's non-stroke side, and then this is the stroke side, which we call this ipsilateral side. All you're seeing is all different time points. And the blue indicates the genes that is decreased, and the red is showing the increase. So there's a 25,000 genes analyzed, and what we come up with is about 822 genes in the non-stroke side is significantly perturbed. On the other hand, the stroke side, we have almost 3,000 genes. This is perturbed at a given time point. Now, this is 3,000 genes in the stroke side. Now, you can put it into the other. How these genes pattern changes over the time. We're talking about from the 1 hour all the way to the 6 months. So as you see here, there is a full cluster really shows these patterns, and out of the 3,000 genes, about 300 genes actually downregulated, which is a cluster 2. And the other 3 are actually increased. So when we looked at their reactants, that means it's the category of their signaling on the, and then what you see is the cluster 2, which is downregulated. This is mostly the nervous system or the signaling. On the other end, the other 3 clusters is cluster 1, 3, 4, which are almost 90% of the gene. And then you actually will see those are the ones, and it really shows the immune system. So this upregulated gene is really represented in the immune cells. Now, this is the gene. And can we actually see this gene of regulation over the different stages of the stroke is related to what cells is coming into the brain. So this is the cell tracking studies. And what we did is, okay, we have 3 groups of mice. And the mice get the stroke, which is middle cerebral lesions. And then there's 3 cohorts. The mice can survive up to 3 days, or 7 days, or 60 days. So each time is representing acute, subacute, and the recovery period. Before we kill, 1 day prior to we kill the animals, and then we actually put the cells that express the green fluorescence protein so that we can track where it goes. So how the cell is going into the brain after stroke. And then this is actually using something called the MCP and CCR2 axis. And this interaction is very important for the cell to go into the brain. So after stroke, that is produced in the injury sites is the MCP1. As it says, this is monocyte chemoattractant to protein 1. And this is looking for their receptors, which is the CCR2. And this CCR2 is not only is the MCP1 receptor, but it's highly expressed in the circulating monocyte. And when they find their MCP1, these are the ones circulating this going into the brain. So basically what we want to say is the green cells in the brain, left side, which is non-stroke, and the right side, in the stroke side. So this is the full cytometer, so we look at the green cells. And then 3 days, 7 days, is 2 months period. Compared to the non-stroke side, which is, as you see in this and the stroke hemisphere, you see a lot more green cells going in. The real surprising fact is, even at 2 months, we put this cell only 1 day, 59 days and 6 days, 24 hours. Still, we see a lot of the cell is going in. That means the cells are continuously going into the brain after stroke. So to visualize it, and then we can actually do the staining of the brain. So this GFP staining, contralateral side versus the lateral side, you see a lot more the green cell is showing here. And not only that, when we are co-stained with the immune marker, which is alpha-1, and now you see the red and green together, this is yellow, and then it actually those green cells are green cells. So far, you see the gene, an immune gene, immune reactor is robust, and persistently expressed across the different stages of stroke. And not only this is accompanied by the immune cell infiltrations. So, the real question is that, is there any way that we can manipulate the peripheral systems? So I'd like to introduce here ischemic conditioning, which was just a top step by the preconditioning. But so basically, when you give the noxious stimulus that it does not harm the organism or does not kill, if you survive it, it makes you much more resilient to the oncoming insult. So idea is that if it doesn't kill you, it makes you strong. Right? So this is an endogenous protective mechanism, and then so we want to look at this this ischemic conditioning. So when do you have to apply, and where would you like to apply this conditioning? So since we are trying to protect the brain, and then you don't want to be the brain, so you want to away from the brain. So the area that we would like to do is the limb, which is the leg or the arm. And then when you want to apply it, because it's preconditioning, therapeutically, we don't know when it's going to have a stroke, so it will be better if we do the posture conditioning. So basically, this is the protocols that what we do is the mice. This is putting in under anesthesia, and then here is a very small blood pressure cuff on the limb, and then basically you actually close or inflate it 20 milliliters each, and in that way you make the limb ischemic for 5 minutes, and then you let it go. So 5-minute inflation, 5-minute deflation, and you have 5 cycles. And then this is our post-stroke limb conditioning protocols. So when we look at this, so Now, we are doing actually, protecting the brain after stroke, but we are putting in the conditioning in the limb. So what would be actually the mediator of it? And then this is obviously we thought this is the immune cell must be involved into this. It's for the RSA or stroke conditioning induced any benefits. So which cell should we study with? In stroke literature, you can actually find the monocyte in the places important role in the stroke-induced inflammation and injury. But this is also important in the stroke repair recovery. So there's this dichotomous role of it. And in fact, if we try to block the incoming the monocyte, the functional recovery does not occur. So we thought it is maybe the immune cell is still probably important. But in monocyte, it is not just the one population, it does have the two distinct populations. So the first one is the pro-inflammatory monocyte, and then this monocyte express in the CCR2 side, and then so that they can attract to the where the MCP or monocyte chemoattractive protein is expressed. And this is eliciting inflammations and very cytotoxic, and then it causes the tissue injury. And the other monocyte is called the anti-inflammatory monocyte. It does not express the CCR2, but it has a tissue repair and remodeling, and that's what the function of this. So using this, so with this, the two subsets, our question is the ischemic conditioning change in monocyte subset? And if it changes, how does it relate to the functional outcome after stroke? So first of all, without any stroke, and we wanted to see whether the monocyte changes by this post-stroke conditioning. No, in this case, it's not a stroke. So we just did the conditioning in the mice. And then this is actually the way that we can identify the other monocyte subsets. So using the flow cytometer, we have identified the other monocyte subset, and then we can have the anti-inflammatory as well as the pro-inflammatory monocyte. And in the blood, we could take it, and then how does it change? Compared to the sham, that means it's just a control. Conditioned animals decrease anti-inflammatory, but increase pro-inflammatory monocyte. And what happens with the other stroked animals? So for stroked animals, the way we did this is we had an MCAO, which is the stroke, and then 2 hours after, and then as we applied limb conditioning for these 5 cycles, and then we looked at on their blood and brain in the 3 days, and then the monocyte changes occurs, and which we saw in the blood. Yes, anti-inflammatory monocyte is reduced, but we did not see the increase of pro-inflammatory monocyte, and this is probably due to that this pro-inflammatory monocyte probably went into the brain. So when we looked at the brain, and then we saw there is the reduction of the anti-inflammatory monocyte, but increase the pro-inflammatory monocyte by conditioning. So it really shows the conditioning in the shift monocyte toward the pro-inflammatory subset in the stroke ones. So how does it relate to the functions? So these animals, we look at, this is the gold standard that people are looking at in the animal model, is injury size. So compared to the control, that is the sham conditioned mice, compared to the size of their injury and limb conditioned mice, they have moderate reduction, nevertheless a significant reduction of injury size and as well as the brain. But really more importantly, when we look at the motor functions, so this is something called the road rod, and the animal was putting it in the rod that is rotating with the increasing speed. When there is a motor deficit, this animal cannot sustain it there. So compared to the before stroke, and then actually you see there was no difference between the conditioned mice or no conditioned mice up to 2 weeks. But about 3 weeks later, and we start to see the differences, and then conditioned mice is doing so much better in the delayed sessions. So one of the mechanisms is that we are trying to figure out, what is the driving of the molecules that actually shows and then these functional benefits. So, one of the obvious choices because is that there is a cellular shift toward the pro-inflammatory monocyte, and the monocyte, this pro-inflammatory monocyte is expressed CCR2, though this is maybe CCR2, this is still one of the target that we can look at it. So how we so what we did is basically we put the animals in the stroke animals, the CCR2 knockout or CCR2 deficient monocytes. And then when we do that, and what we expected the monocyte shifted toward the pro-inflammatory, there was no difference. And then in terms of the function, there was no differences. So basically, what we saw the pro-inflammatory monocyte shift, as well as functional benefit, was disappeared in the absence of the CCR2. So at this point, that I guess CCR2 is a mechanism, and then the limb conditioning is really gives the monocyte shift and then also functional benefit in the delayed effects. Still, it doesn't answer why we gave this conditioning at 2 hours after stroke. And as you see, the actually functional benefits is actually started somewhere around the 3 weeks and is sustained. So what is the underlying mechanisms that this delayed improvement and the motor functions after conditioning? So after stroke, and we are really looking at where the injury is, but this injury is for the brain in the stroke side. They are continually get the atrophy. So I wanted to see whether stroke-induced brain atrophy is maybe related to this delayed motor functions. So striatum is the one that is the major impacted area by middle cerebral atrophy. So if you look at the striatum, and then this is the volume analysis, and then compared to the non-stroke side, which is over the time, there really doesn't change much. The stroke side, which is the red line, about 3 days it goes up. It's probably due to the brain swelling. But by 1 week, it gets smaller. But even after 1 week, it's continually getting smaller and smaller until 2 months. And maybe 6 months around it is stabilized, maybe the fibrotic scar at the time. But striatum has also connected to other areas. One of the other areas is, so this is the mouse brain after 6 months, and we can actually appreciate how much the atrophy has been occurred in 6 months. So, because the striatum is connected in a place like venous alluminous and the substantia nigra, we thought maybe there's a continuous the secondary injuries going after stroke. So if you look at the alluminous area compared to the contralateral side, the ipsilateral side, this is going down, but it's significant around 2 months and 6 months. And then one thing that I didn't realize is the alluminous, even contralateral side, they're going down around 6 months. So this is the age-dependent, maybe atrophy, even healthy contralateral side. So that was just a big structure. So we can actually do the volume analysis. But the substantia nigra, it is so small, we really cannot do the volume analysis. So we we decided to use the other animals that express the green fluorescent proteins in the dopaminergic neuron, which is the TH. So we're using the TH mice. So TH is for the tyrosine hydroxylase, and this is regulating enzymes in the dopaminergic neurons. What you see is when there's a stroke in the striatum, and then this cells in the substation diagram, dopamine neurons, is the loss in after stroke. So using these lines, so what we have been done is we clear the brains, and we actually look at how they change. But because of the TH, GLP mice, we can actually see all the TH cells in this brain. So you actually see the hippocampus just to show, and then now you see the striatum, and which is here, and then you see the much smaller and the hippocampus, and the striatum is showing to the upper part is the where the stroke is done, and then you now see the beautiful the substation diagram in the whole brain. So let's get back to the conditioning. So when you do the conditioning, what happens? So the clear the image, this is the compact the images, this is contralateral side and ipsilateral, and this is 2 month after stroke. And all those dots are the TH neurons, TH expressing neurons. And substation diagram is compare the contralateral side and the ipsilateral side after the stroke, and the concept disappeared. And okay, and when we did the ischemic conditionings, and then you actually have to rescue some of them, but not in a whole lot. So we also used the very classical method, just cut the substantial nigra areas to see, and then compare to the sham mice, which is about lost about 50% of the TH cells. And the conditioning animals, they actually was attenuated the TH loss. So this is the one last the slide, which I got the last night. So I was trying to insert these movies. So basically, they block the substantial nigra regions. Let's see whether we can see. So all those dots are the TH neurons, because this is the way we can actually unbiased way to see the TH neural death. The blue is the posterior, and then the yellow, and actually red is the anterior. So if you do composite, actually this is the way to see, and then compare the contralateral side, this from the rostral corals, then we actually see the reduction. So we also looked at the so also we looked at this one of the sections is looking at the immune cell markers. So when there is the here is normal brain, but here is a lot of TH cell is lost, and then you can actually see a lot of the immune cell is associated with the cell loss. So to summarize that stroke, there is persistent immune gene expressions, which is accompanied by the immune cell infiltrations, and then there is a tissue loss, and the continuous tissue loss, not only in the primary injury site, but also remote areas. And then this infiltrating the peripheral immune cell is associated with the secondary degenerations as well. And then when we apply the remodeling conditioning, it shift in the monocyte from end-time to the pro-inflammatory monocyte, and that shift is associated with a better functional outcome, and especially the motor function in the delayed sessions. And we identified the CCR2, this is one of the key mediators of it. And then also RSA is my primary, preliminary data, at least shows that this attenuated TH neural loss at the 2 month post-stroke. So really the big picture of this is that we can manipulate the peripheral immune systems to be able to change or even promote and the pathology as well as the enhance in the long-term stroke outcomes. And at the end, the post-stroke conditioning probably can be used as the immune-based therapies to improve and stroke recovery. So with that, we have the lab members, the role-based people, those are the ones who directly involved in this particular project. And then John, now Institute is to provide THGFP mites and for us to straddle minor pathways. And the Regeneron Pharmaceutical Company, Susan and Wayne, really done that analysis of the transcriptome and then zoom out from the Rockefellers, the brain imaging, and then versus funding from the NIH. Most importantly, Burke Foundations. That without that, I would not be able to sustain and secure the other fundings. So, this is my
So that was fascinating work. I'm interested in the contralateral thalamus decrease in volume. Do you see that in mice over a 6-month period?
Yes, that's what
in control mice.
Of normal mice?
Yes.
I guess there will be, because this is a 6 months, they are about 10-month-old. So, I mean, when how normal aging, we actually see the atrophy. The reason striatum does not change is because there's something ongoing on the stroke side. Maybe that's why they are still there, not much of a difference. But thalamus, this is not the area that is primarily impacted. And then the contralateral side is actually decreasing this. So,
it would be interesting to actually compare it to the control mice, because in fact in humans, you know, there's been a lot of work saying that a stroke can actually sort of induce or potentiate dementia. So we see people who fail to recover, in fact, get worse after stroke, have more atrophy in the contralateral hemisphere, in the cortex, than controls over a 6-month period. So we think that that's what's happening, is the stroke's actually inducing neurodegeneration. So it'd be interesting just to compare controls as well as the
I think the
control side.
analysis, I think this is pretty interesting. Yes.
Great. Yeah.
Yeah. So you, if I got it right, you showed an increase.
Susan, could you repeat the question?
Yeah. You showed an increase in the pro-inflammatory monocytes by OI60 high. By low. So are you able to compare the transcriptome profile of those to the ones in the absence of the hindlimb ischemic? So I'm wondering if it's just a number issue or if it's some quality issue. Like, what do you?
So that was a full-size terminal, right?
Yeah.
But transcriptome, we have not done that, but that was the marker by LI60 high and low, and then we testing between the it's not a change, it's just a
right. I don't know if you get enough cells back to do that.
That would be really interesting that we could do that, yes. But we have not done it.
Okay, I think we'll move on, so we're running late. Our next speaker is our final speaker of the day, also part of the home team, Tago.
Thank you.
So some support from the Peanut Gallery, who runs our human motor recovery lab, particularly interested in stroke. And Tomoko joined us a little over a year ago in November, and has really hit the ground running. And I think you'll be very interested to hear a lot of what she has to say about stroke recovery and the motor system.
Yes, well, thank you so much for staying until the end. I'm really honored to be part of this distinguished speaker panel and getting to meet all of you women leaders in stroke. It's been a really great opportunity for me. So I'd like to share some of the work that we've been doing in our lab on motor recovery after stroke. So as you all know, stroke is a growing problem in the United States. It affects 800,000 patients per year. And the costs related to stroke care and the loss of productivity due to stroke reach upwards of $34 billion. And despite this enormous cost, stroke remains a leading cause of chronic disability. And then one of the reasons that we've chosen to study the motor system in our lab is that the majority of patients have hemiparesis or weakness after a stroke. And when we look at patients who are 6 months after their stroke, so this is after a period of recovery, we'll find that 50% still have some degree of hemiparesis. Right? And those motor deficits then contribute to the 30% who are unable to walk without assistance, the 26% who are still dependent in their activities of daily living. So first I'd like to take a look at how a stroke affects a given individual. So this is taken from the World Health Organization ICF framework. First, we have impairments of bodily functions or structures. An example of this is hemiparesis. Because of those impairments, then we have activity limitations. So the inability to perform certain tasks after a stroke. For example, not being able to walk. And then participation refers to how one is functioning as a member of society. So the ability to work due to the stroke. So for a multitude of reasons, the goal of clinical rehabilitation has traditionally been to treat limitations in activity. This is rehabilitation as it's delivered by therapists in hospitals and in clinics. And they do this by teaching strategies to work with the patient's level of impairment that they already have. So some examples of this, maybe if you have a stroke that affects your dominant arm, they may teach you to do activities like feeding yourself using the unaffected side. Or if you have trouble walking due to loss of balance or weakness, they may teach you to safely ambulate with a cane. So in contrast, the goal of our research in motor recovery here at Burke and elsewhere is to tackle this problem at the level of impairment. So that means that we want to get patients moving the way that they used to do before they had their stroke. And this likely occurred through a process of neurorestoration or repairing the brain. So these two strategies of neurorestoration and clinical rehabilitation are really synergistic. As clinicians and researchers, we have a shared goal to really help our patients to move better, to achieve independence, and really to cure stroke-related disabilities. So I want to take a step back and to look at what we know already about recovery of motor impairment in the upper limb. So this is a study that came out when I was a neurology resident at Columbia, looking at motor recovery from in the first 3 months after the stroke. So the authors of this paper looked at motor recovery as the change in impairment from the initial period, so in the first 24 to 72 hours after their stroke, and again at 90 days. And the way that they measured impairment was using the Spiegel-Myers scale. So for those of you who aren't familiar with this, it's a widely used measure of motor impairment. It consists of 22 items for the upper limb. These are some examples of activities that are tested, so ranging from coordinated movements of the entire limb to isolated movements of the wrist and the hand. The scores on the scale range from 0 to 66, with 66 being the best possible score. So interestingly, what they found was that motor recovery was proportional to the initial impairment. It actually was equal to 70% of the patient's maximum potential recovery. And another way to say this is that patients tended to regain about 70% of what they had lost on the scale. Let me show you 2 examples. So you have 2 patients here at admission in 3 months. Patient 1 starts off moderately impaired with a Spiegel-Myers scale of 4 of 36, and their maximum potential of possible recovery becomes 30. If they then regain 70% of that, they end up with a score of 57. Patient number 2 starts off more severely impaired, so Spiegel-Myers score of 10. They now have a greater maximum potential recovery. Again, if they've achieved 70% of that maximum potential recovery, they end up with a Spiegel-Myers score of 57. So now this predictive model has been validated in multiple patient cohorts. I have to mention that there is a subset of severely impaired patients initially that don't follow this rule, who are non-recoverers, and I won't get into this today, but it does appear that you need to have some residual cortical spinal tract integrity in order to exhibit a proportional recovery. People have also shown that this proportional recovery is not influenced by the amount of therapy that people are getting for the upper limb. So taken together, these results suggest that the early motor recovery that we see, at least at the level of impairment, is due to spontaneous recovery processes. We then wanted to ask, what is the time course of spontaneous motor recovery in humans? I think it's important to really understand when that recovery is occurring, because you may have interventions that are targeting the acute period, the period of recovery, and then the period beyond that period, that spontaneous recovery. I'm going to show you some data from the SMART study, which was a multi-center longitudinal observational study in which we followed patients' recovery over the first year after their stroke. In addition to the Feeble-Myers scale, we decided to measure specific motor impairments. And as we were thinking about developing these tests that we wanted to measure, we wanted to make sure that the tools were objective, that they were sensitive, they were quantitative, and had no floor or ceiling effects, which are some of the limitations of the Feeble-Myers scale. These are two examples of the types of impairments that we want to follow. So one is proximal arm coordination, so the ability to move your shoulder and your elbow in a coordinated manner. The second was finger dividuation, so the ability to move your fingers independently. And these two types of motor control are important for all of the complex skilled movements that we perform every day with our arm. So this is an example of the device that we use to test this. This is in our lab. All of you are welcome to come and play with it if you'd like. So we have subjects seated at a laptop table. Their arms are fully supported on air slip-like devices to minimize effective friction. It feels a little bit like you're playing air hockey. And this is the very simple task that they're asked to perform. So they have a cursor that's corresponding to their hand position. They're asked to move it to targets as they appear. It's really a very basic test of your ability to control your arm. Now, this is an example of the type of data that we get from this task. These are movements that are made on the left by a healthy volunteer. On the right are the movements made by a patient with stroke. And you can see very obvious differences between the two. Right? So the stroke patient is making very variable movements. They're not always hitting the target. They're curved. And one of the challenges of working with this type of data is that you may find yourself interested in looking at 5 different variables, 10, maybe even 20 different variables. And what we wanted to do was to try to come up with a way to quantify the differences in these movements with an overall score. So to try to capture what we see visually in a single score. So over the past few years, we've been working with a biostatistics group down at Columbia, led by Jeff Goldsmith, and he's developed a functional principal components analysis of these reaching movements. I won't get into the details, but basically the way it works is you can describe these movements in terms of these components and put different weights on them. And this allows us to arrive at an overall trajectory error score. So this looks like visually is that, say, the movements in gray are what the patient is making. You want to see how much they deviate from that red line. So in this SMART study, we tracked patients' performance on this task over the first year after their stroke, starting from the first 2 weeks out to a year. And what we found, so here we have time after stroke, and then this trajectory score. So lower scores are better because it's measuring how the distance between what a healthy control reference population is doing. You see that over the first 2 time points is really where you see the most improvement. And after 5 weeks, we see very little changes in this measure. And it's not because there's a floor effect of this task, because as you can see, compared to the affected arm performance of compared to unaffected arms, the performance of the unaffected arm is much worse. We also, were able to add another type of motor control. So we had a task that measured finger individuation. So what the patients place their fingers on this device, and under each finger there's a force transducer. They looked at a display, and the green bar corresponding to the finger lit up, the rest is pressed down with that finger only. And this is a task that's very difficult for patients. So as they're pressing down, all the other fingers tend to become active. And from this data, we derived what we call an individuation index. When we plot this measure over time, over the first year after stroke, we see that, again, recovery occurs fairly early on. Here we see recovery up to about 12 weeks after stroke, and not so much thereafter. So in summary, we showed that the recovery of motor impairments in patients after stroke occurs really early on, so somewhere between 1 to 3 months. But as I showed you in the first slide, 50% of patients still have hemiparesis at 6 months after their stroke. Right? In the US, there's currently 7 million stroke survivors with chronic disability. So the challenge is, what can we do for these patients who already experienced the spontaneous recovery that they will have and reach this plateau? So one potential way of looking at this problem is by thinking about plasticity, which we'll broadly define as the ability of the brain to change, either in response to injury or to experience. We know that plasticity is something that we all have. It allows us to learn new skills, even in the healthy brain. Acutely after stroke, we've heard a lot about all the different repair processes, the new processes that become active, leading to an environment of hyperplasticity. But what about the chronic post-stroke brain? We actually don't know much about plasticity in this population because it hasn't been well studied. However, there's a thought that perhaps, at least relative to the acute period, that the plasticity is lessened. And the question becomes, can we promote motor recovery in the chronic stage of stroke with drugs that enhance plasticity? There have been a number of exciting results from preclinical studies about drugs for stroke recovery. However, currently there are no FDA-approved medications for recovery. And one of the challenges, I think, in developing drugs for use in humans is that we don't have an easy established method to determine that a drug reaches the central nervous system and has any effect on plasticity. One potential approach that we've been exploring in our lab is the use of this technique called PAS, or paired associative stimulation. So this is a non-invasive way to induce plasticity in humans. It consists of pairing a peripheral nerve stimulation, a sensory signal, with transcranial magnetic stimulation, which I'll describe in the next slide. It relies on the relative timing of these two inputs to modulate cortical spinal excitability. And this is considered to involve an LTP-like process, which is important for learning and memory. So this is what the transcranial magnetic stimulation setup looks like. The subject just doesn't have to do anything. They're just seated in their chair. Basic equipment consists of a stimulator attached to a coiled wire. And when the stimulator is triggered, it sends a brief electrical current through the coil, which induces a magnetic field that can then penetrate through the subject's scalp and the skull and stimulate the underlying cortex. So when this TMS is delivered over the primary motor cortex, it can evoke a muscle twitch in the contralateral hand. And this is measured with EMG sensors, and it's called an MEP, or motor evoked potential. So we asked the question whether we could use this technique to induce plasticity, measure plasticity in patients with chronic stroke. This technique was first developed in healthy volunteers, in which it worked pretty well. So this was the paradigm. So patients underwent PAS, and before and after this PAS intervention, we tested the excitability of their brain using TMS. So what we found is that comparing before to after TMS, we saw that we were able to successfully induce plasticity or this increase in this motor evoked potential. We then took it a step further and asked whether PAS could be used as a tool to assay the effects of drugs on the brain in chronic stroke patients. So this was a study that we did in collaboration with Dr. Dobkin, who is one of today's awardees, in which we looked at the effect of memantine on PAS. So memantine is currently approved for the treatment of Alzheimer's disease. Its mechanism of action is a low affinity, non-competitive NMDA antagonist. You've already heard a bit about the NMDA receptor today, but the synaptic NMDA receptors are really critical for normal synaptic transmission and facilitating plasticity, and it's involved in LTP learning and memory. However, there are also extrasynaptic NMDA receptors, and these have really been implicated in pathological conditions, including stroke, and they can actually have a detrimental effect on synaptic plasticity. In preclinical studies, it's shown that low doses of memantine can selectively block these extrasynaptic receptors, but spare the synaptic receptors, so they're allowing for normal synaptic transmission. So our hypothesis for the study was that memantine had a dose-dependent effect on plasticity due to differential effects on these extrasynaptic and synaptic NMDA receptors. And we used a range of doses that are used clinically for Alzheimer's, so between 5 milligrams and 20 milligrams. Our prediction was that with 5 milligrams at a low dose, we would have enhancement of plasticity, whereas with 20 milligrams, you would block both the synaptic and extrasynaptic receptors, and therefore block this plasticity. This was a study design, so the baseline session, people were tested to see whether they had a PAS response, which they did. And then each week, they received memantine at increasing doses. And on the 7th day of that particular dose, they came back to the lab and were tested to see whether what happened with the response to PAS. So contrary to our hypothesis, where we predicted that there would be a differential effect of dose, we saw that memantine actually abolishes PAS-induced plasticity at all doses that were tested, including 5 milligrams. So, unfortunately, this didn't seem to work. So, we would say that it's unlikely that memantine, at least at these doses, would be beneficial for stroke recovery, at least in the chronic stroke population. So just to summarize, so we were able to show that this is a PAS is a tool that can successfully look at plasticity in patients even with chronic stroke. It can demonstrate CNS system engagement by drugs. As I said, we weren't able to show that it enhanced plasticity at any of the doses that we tested. However, we're envisioning using PAS to test other drugs that have potential effects on plasticity. So in conclusion, I want to leave you with some thoughts. I think as researchers, we need to focus our efforts on impairment rather than activity, because I think that impairment really reflects the ongoing activity in the brain much more so than looking at forms of different tasks. We also now have the tools to really examine motor impairment, whether it's in the context of spontaneous recovery or in the response to neurorestorative interventions. We also have the tools to determine whether drugs have an effect on the brain. And In the future, we hope to combine these drugs with impairment-focused training to really promote motor recovery and help our patients recover better. So with that, I'd like to acknowledge all of my lab members and collaborators. You can see from this extensive list that it really takes a lot of collaborators to get this kind of clinical research done. I have a lot to thank the funding organizations, including the Burke Foundation. Also, I see some patients in the audience here. I'd also like to really thank you all for your participation in our clinical research studies. Without you, all of this wouldn't be possible.
We have time for one or two questions. Last parting comments. Great job, Tomoko. I really want to end by thanking. Was there a question? Sorry.
Sure. Do you have any idea about how much of a change in PIS you need to see to have an outcome in terms of the impairment? So, how sensitive is it? How much of a change is reflective of a change in impairment? Do you have a figure, sort of a feeling on the nuance?
That's a great question. I think there's still a lot of work to be done with this technique. We've been successfully able to induce plasticity with this neurophysiological measure. There's still work that's being done looking at the correlations between this kind of PS-induced plasticity and motor learning and performance of motor tasks. So I think there's still a lot of room for experiments that need to be done.
Yes, another question.
I have a question regarding some basics, because I believe that it's very important that preclinical research and clinical research should have some uniformity in terms of outcomes and model stuff.
Yeah.
The thing is, first question is, do you also see the proportional recovery in these more sophisticated methods? Or is it just only observed using the Kugelmeyer?
So far, it's in the motor domain. It's only been reported using the Kugelmeyer score. I think we're hoping to collect enough data with using these kinematic tools to be able to answer your question. Right now, we don't have enough data to really model those well.
Second question is, the patients are doing very well, which probably shows me that the lesion sizes are small, or a huge number of the patients they have small white matter lesions. What can you tell about lesion characteristics of the patients?
Are you referring to the proportional recovery?
No, just, so I believe so that, or let me ask you a question. Yeah. If I want to study forearm recovery in an animal model, what kind of stroke model should I use? So that I can really mimic your patient.
Because of various exclusion-inclusion criteria that are used for these studies, these are fairly complex motor tests that are being asked of these subjects. So they tend to be those with smaller lesions. They can be portable, but not involving entire primary motor cortex. A lot of them are subcortical, so internal capsule or brainstem. But we do have somewhat of a wide range, maybe not the hollow middle cerebral artery.
So you could use a smaller distal cortical model, like some of the ones we were showing are those large MCA, that you can do either a transient or a permanent distal MCA, and it gives you a very nice small cortical infarct. It's just not something we see that often in patients, but you could also do a white matter lesion, but then it's difficult to do through a suture. You can do that with the bilateral, with a carotid artery stenosis model. Sometimes you'll get some motor deficits that might be more like yours if they're white matter.
Mm-hmm.
Great. Thank you. this is part of an ongoing dialogue, so if you have questions or suggestions, email us. Have a great weekend.
Do you have any ideas?
I think probably.
Thanks. Yeah, thanks.
Well,
Great.
Perfect.
Thanks for letting me.
Yeah, thank you.
Oh my gosh, all the time, full empire. It really is.
That's right. So if you're prepared to have a great weekend, all this Christie,
All right, thank you.
so I think for her, not okay, because you'll have to think about the timing. There are very few bioavailability
She talked about,
yeah,
but she talked about the microbiome. She talked about the 5.
you may have non-responders.
Like that.